And so with that, I think we can move into today's meeting. I think I've gone through most of the welcome, and we can move into the first personal perspective. But just as a review, today we have a session that is mainly looking at how different groups have built solutions and utilizing some innovative drug development tools through their collaborations. And so we have 5 presentations from a breadth of people who have worked in different consortia or in groups within their research area. We did decide to shift the panel discussion time and the breakout session times a little bit. So the panel discussion will be a little bit shorter. Will be shorter today to permit more breakout session time. And I think that everyone will appreciate that piece, especially since the second day, and I know that we have an overlapping workshop. There may be fewer people during today's session, and so that will ideally permit even broader and better engagement within each one of the breakouts. A short break, and then we come back and look at some of how Cpath and other areas has overcome barriers to data sharing. And then 2 presentations around data sharing for solutions and with perspectives from an individual, a scientist at the FDA and a scientist from industry. And then we will finish the day with one last panel discussion, and then I will close out with a summary and a call to action. So with that, we can move into our first personal perspective, and there'll be one by Kevin Fowler and by Richard Knight. And so let's go ahead and switch to the first video.
My name is Kevin Fowler, and my connection to kidney disease is actually over several generations. In our family, we've had multiple generations impacted by polycystic kidney disease. And actually, myself, I was impacted as well. I was actually the first person in our family to receive a kidney transplant back in 2004. And one of the areas that really I have not really learned about until now is this whole aspect about DIKI in terms of education and drug safety, and just also I think just the why this is such an important area to share data, because if you look at what has happened to polycystic kidney disease, you know, that was a disease when I was growing up as a child. There was absolutely a complete black cloud around that because there was no treatments available. In fact, there was very little known about the disease. And now, you know, at this point in life, to see what's happening because of data sharing and because of people working together, there's now one treatment approved for the treatment of polycystic kidney disease, and there's potentially many more, hopefully, in the pipeline. And so I think that just gives a great example of hope and the benefits of collaborating together for the common good and really collaborating for the patient community. Everyone talks about they're all patient-centered, but if you're really patient-centered, we should be doing data sharing, because at the end of the day, it is the patient's data. And for people out there that have kidney disease, I would like them to understand the importance of data sharing, how that can lead to further innovation, and how people should speak up and make sure that this data is shared, because at the end of the day, the data is ours. And we have a say on this data being shared, because I think we all have a desire to have a better future for people to preserve their kidney health. I'd like the research community to understand one thing clearly about data sharing, is that there's a need to educate patients on data sharing so that they understand that data is theirs and that they have a voice in advocating for this data to be shared to solve problems in the kidney disease community. One thing I like the DIKI research community to know is that there is a sense of urgency on this. We've operated in the United States with a kidney care system that's rewarded kidney failure. And now we're moving towards kidney health. And this is one leg to help achieve that. And it's long overdue to bring about changes so that we have a system of care that rewards kidney health rather than kidney failure.
Hello, everyone. My name is Richard Knight. I'm the current president of the American Association of Kidney Patients, largest independent patient organization in the United States. And I am a kidney patient. I'm a 15 plus years out on my transplant. And I spent 2+ years on dialysis. I've got a history and had a history with my kidney disease. And that is part of what motivates me to participate in this workshop that we are currently having. I'm unfortunately one of those that was not properly diagnosed, and I crashed into dialysis. Spending a couple of years on dialysis, I was fortunate that I was able to locate someone who chose to give me the gift of life, a kidney. I had my transplant, and since then, I've lived a very good quality of life. But unfortunately, everyone doesn't have the same good fortune that I have. And that's why I'm very interested and supportive of this workshop, in fact, that we're having today. One of the challenges that we have, and it's certainly been made even more clearer during the pandemic, is that kidney injury, drug-induced kidney injury, is something that is of concern to the kidney patient community. Drug-induced kidney injury has impacted me in the past. When I was in the hospital for pneumonia a couple of years ago, I was given some medication, and I wasn't exactly sure of the impact on me. While I believed that it was helpful for the pneumonia, I was very concerned about the impact on my kidneys. I was given some information, but it really did not satisfy me enough. So I think that we ought to have a better idea about the specific impact of all medications on kidney patients, particularly when you're in there for something that may be non-related to your kidneys. And I'm speaking to my friends and colleagues in the research community. I think that it's very important that we get together through means of collaboration, and we focus on coming up with better information. I'm going to refer to it as biomarkers, so that we can be a little bit more specific and targeted with respect to the diagnostic capabilities, and more importantly, predictive capabilities, so that we can have a better idea and way to determine the impact that certain medications will have on patients, and particularly as we look at the subpopulations of certain patients. Innovation is great, but we want to be certain that as we innovate, we clearly understand all of the implications of that innovation, in this case, different drugs. We understand the impact that those drugs may have on our kidney patients. One of the things I want to emphasize to the DIKI workshop community is that one of the challenges that we have in healthcare is these artificial silos that we have created. We may have silos between our academics, our universities, between our patients, between different industry players. Collaboration, I believe, is the way to go. And I believe that this strategy of collaborating, sharing data, working together, having patient input, I believe all of that is necessary for us to be able to go forward in a healthcare industry that is changing quite rapidly, but we always want to be able to involve all of the key stakeholders. And I think that it's important that we continue to understand the importance of involvement of all stakeholders in the process of developing different innovations and medications for patients. For my fellow patients, I think that it's very important that we participate. We participate in efforts like this. Will your data be shared? Yes, it will, but it is going to be de-identified. And by that, I mean that there will be no connection between your data and you as an individual. But more importantly, the results, the improvement, particularly if you happen to have any type of kidney disease that can be passed on from generation to generation, this is an opportunity for you to participate and come up with the type of biomarkers that can really help determine how a certain disease may be developing or exactly what it is. So I think that I'm going to recommend to all patients, don't be afraid, and you should look forward to participating in clinical trials and other research that's going to allow for the possibility of innovation with respect to medications and drugs that are available to all patients.
Thank you very much, Kevin and Richard, for taking the time to put your message out here for all of us. We do appreciate that and your participation in the workshop. So now we wanted to give a little bit of the highlights from our breakout session yesterday towards the end of the day. And so let's go to the next the slide after this. Okay, so in case you weren't here in attendance yesterday, we posed one overarching question to each breakout, and that was what needs to be done to advance novel kidney injury biomarker application and implementation. And so within this, we asked everyone to take it into account the following 4 questions. What breadth of demographics are needed for meaningful implementation? And so I think by that we mean different disease populations, right? The standard demographics of age, ethnicity, race, gender, will all fall underneath that category, as well as suggestions on pieces of size of number of subjects that may be needed in order to have a good set of baseline values and then potentially an injury changes in following injury. The second was what is the best way to integrate disparate data? And the third, what types of injury need to be assessed? And here we're thinking of different types of kidney injury. And you'll see that in the results in a moment. And then the fourth, what can be done to enable stronger patient engagement? And so I collected the responses, let's go to the next slide, from each question, and then pared them down to what I saw as the common themes across each one. And so, as an example here, we had, it's important to have gender and ethnic representation, more enrichment of those data to understand socioeconomic status. It was also important to understand, have a good understanding across ages, particularly in older or geriatric patient populations. And in the emergency room, a majority of people who present emergency room, we heard from one of the physicians, are over 60, 70, 80 years old. And that doesn't necessarily always fall into the standard clinical trial population and demographics. So expanding our understanding across pediatrics and geriatric populations is important. Looking at utility of different disease states such as diabetes, heart failure, where there may be different baselines. And then additionally, that depending upon how we collect the data, it may require a very large sample size for each demographic. And so, coming out of that, it's going to require equal gender and ethnic racial representation. Age ranges beyond the standard clinical trial are important. And then it would require a large sample size for each demographic. And so when we move into today's breakout, what I would like everyone to begin focusing on is what's on the right half of this screen and looking at ways to address that in a framework for an action plan. Let's go to the next one. So for the next question is, what is the best way to integrate disparate data? We saw here, a mixture of questions and ideas. ranging from sources of data to collect from, as well as components of what is needed to make integrating the data important. And so, here a mixture of clinical trial and real-world data was seen as important. Establishing some standard case report form templates for data transfer, as well as establishing a standardized informed consent that can be used in various settings, that informs the patient of what their data is going to be used for and how it might be used in the future. We here at Cpath, we have a nice publication from one of our other consortia that looks at ways to implement an informed consent in a slightly broader manner to permit research on novel biomarkers, and that's something that we could consider implementing in emergency rooms when patients come in presenting with relevant injuries or diseases that we'd like to capture. And then as well would be leveraging groups that are already collecting samples. One example was the Toxicology Investigators Consortium, which has a fairly broad reach into a variety of demographics that are not traditionally collected in a lot of our phase 1 clinical trials. They have reach into different communities in the US. They have, I think, strong collaborations and working on some of our Native American land as well in different communities throughout the US. So those are some of the suggestions that we're seeing here. And let's go to the next question. So what types of injury need to be assessed? And here we begin to see, looking at the kidney-specific regions. Kind of the theme that came up is different sites of kidney injuries, types of injury, mechanism of kidney injury, and then again, establishing standardized informed consent to use here, and some desire to have integrated data available worldwide. And so this one, I think we had some very specific asks in terms of the types of injuries. So that does assist in moving forward. Let's go to the next slide. And then, what can be done for stronger patient engagement? And here we had a number of responses, and kind of pulling out from those, what I saw as themes are: one, articulate the research so that patients see its impact on the patient's life, and then as well include patients and advocacy groups in workshops similar to this, where we're talking moving into some of the science and regulatory decision making. Utilize patient advisory committees, and then I think an assessment within companies is asking, continuing to ask what companies can do to make patients feel safe about data sharing. And so that our presentation on different data sharing successes. We did have a few questions that popped up at the end, and I have that on the last slide. Yeah, so one question: has it been determined that the biomarkers that we currently have proposed are sufficient? And they may not be. I mean, there's the biomarkers that within PSTC have been assessing are all primarily tubular injury biomarkers, and so there may be a need to look at biomarkers that are looking more at the glomerulus or at causes, pre-renal causes of kidney injury or post renal effects as well. And so those may be some areas that we branch out into, and there are some nice publications that propose different biomarkers for different regions of the kidney. And so that's something that can definitely be explored in the future. And then as well, what can companies do to make patients feel safe about data sharing? And I think that's an excellent question that we should be asking each time we're looking at contributing data and collecting data from individuals at the beginning. And so with those items, I would like to move into our next section. And here we have a nice breadth of presentations looking at success through collaboration. These aren't solely looking at kidney injury, though the majority of them are. And so we begin with Dr. Chirag Parikh, who is a professor of medicine and the director of the Division of Nephrology at the Johns Hopkins University School of Medicine. And so with that, Chirag, we will stop sharing our slides and permit you to bring up your slides.
Can everybody see this?
No, not yet.
Oh, not yet. Oh, I thought I did it.
There, now it's beginning to show up. We see your slides. They aren't in presentation, but there we are. Perfect.
Perfect. So thank you, Nick, and thank you, Cpath and everybody for organizing this. I'm delighted to present this work as an example of collaboration that happened between various institutions and development of biomarkers for drug-induced AIN. And a lot of the work that I'm presenting here was done by my mentee, Dennis Muladina, who's in the audience here. And these are the main studies where the work is presented from. Here are my disclosures, but the main one for this talk is Dennis and I are named co-inventors on the methods for diagnosing acute interstitial nephritis. So for people in the audience who do not get to see what is AIN, this is how the kidney looks under the microscope with AIN. The picture on the left is a healthy kidney with normal tubulointerstitium, and the kidney on the right shows infiltrate, eosinophils, macrophages, and T cells. And when the AIN advances, you also get tubulitis, or what is called as injury. Please pardon my voice. I'm recovering from a bronchitis. So what is AIN? And I think yesterday, Jonathan Himmelfarb presented a case, and Dr. Bonventry gave some idea that AIN is responsible for about 70% of the drug-induced injury to the kidney, but it's characterized by presence of inflammatory cells. This is distribution by age group, but we see that within AIN, 70% of the cases are because of the drugs, and the common drugs are NSAIDs, amoxicillin, antibiotics, PPIs, and more recently, immune checkpoint inhibitors are becoming very common causes of AIN. About 1 million people receive these agents for cancer, and it's becoming a very common cause of clinical consult. However, another important point is if you are age greater than 65, chances that you have autoimmune causes of AIN, which are Sjogren's and other diseases, is very low. It happens in younger patients. So in older patient, AIN is usually because of drugs. Here is the prevalence. If you take kidney biopsy registries, about 20% of patients have AKI. For this conference, I looked up the KPMP study where we have the AKI arm. And out of the 35 biopsies that we completed, and these were all research biopsies, just to give you an idea, 48% patients had drug-associated injury. This number was surprising to me. I was expecting 20 to 30%. And the common causes after clinical adjudication by our team at KPMP, independent nephrologists at various sites, was vancomycin antibiotic, NSAIDs, and we had several other drugs that were responsible for these cases. The key part is AIN is a progressive disease. If it is not treated, it's treatable. You can give steroids, you can take the drugs away, but if you don't treat it, it results in CKD and ESRD. And this is the expected incidence in USRDS data sets. So the few key concepts for AIN is the diagnosis is challenging. Nephrologists have to keep a very high degree of clinical suspicion as there is no specific sign or symptom. It's sometimes a slowly insidious disease, happens over weeks to months. 50% of the cases present as AKI, the other 50% presents as acute kidney disease. And there is no currently available clinical test which is pathognomonic of AIN. So we are, as nephrologists, we resort to kidney biopsy for diagnosis. This is done at academic centers, but if you are in a non-academic setting, biopsy is very difficult to set up and get it done. Even if you wanted to get a biopsy done, it's not for everybody. Patients who have high risk of bleeding or for some reason cannot get it done because they are ineligible, they do not get a biopsy and the diagnosis is delayed. And once again, the disease is reversible if identified and treated in a timely manner. So to develop biomarkers for this disease, this is what Dennis and I put together. This is the study flowchart where we enrolled over 200 patients getting kidney biopsies, collected blood and urine samples at the time of kidney biopsy. And the causes for kidney biopsy were unexplained acute kidney disease. All the biopsies were adjudicated by 3 independent pathologists. 15% participants had AIN, the rest did not have, had all sorts of other conditions. And since it is a 10-minute talk, I'll focus on 3 aspects of when I show you the results of biomarker development. When we were developing and selecting biomarkers to advance, we focused on making sure that the assays were clinically and analytically valid. This includes ruggedness, robustness, pre-handling of the samples, etc. Whether they have clinical validity, high values in the disease patients, as well as associations and clinical utility, where we think about prognostication, prediction, and discrimination. So we tested over a dozen biomarkers from the immune pathway, Th1, Th2, Th17, etc. But the 2 biomarkers that emerged as good for AIN were urine TNF-alpha and urine IL-9 from Th9 pathway. The levels were much higher in AIN compared to all the other conditions such as ATN, GNs, DKD, and normal patients who we compared as healthy references. And these were the conditions that you get when you biopsy patients with unexplained rise. The commonest is AIN. We initially discovered these biomarkers in the first sub-cohort that we enrolled between 2015 and 2016, but we continued enrollment and subsequently demonstrated that the results are remain strong in the same sites where we continued enrollment. We have now externally validated these findings in other cohorts from C-Probe and Neptune, as well as from Indiana and Johns Hopkins. Just to show you the consistency of evidence, when we look at the histological parameters and compare the biomarkers, we see that with worsening interstitial infiltrate score or worsening interstitial eosinophil, the dose response pattern is present with the biomarker levels in the urine. Same thing with tubulitis. And we wanted something specific and that is different from acute kidney injury biomarkers. And to our delight, when we tried to see how they did with ATI on kidney biopsy, there was no association showing that these biomarkers are picking up the immune mediated injury in the kidney. And once again, we did some more tissue interrogation and were able to confirm in the kidney biopsies that both these proteins, TNF-alpha or their receptors, FCER1, were present in the AIN cases and had correlation with the levels in the urine. When we move on to clinical validity, we wanted to demonstrate they are independently associated with, after adjusting for a lot of clinical parameters, we adjusted the biomarkers for each other as well as for other clinical markers such as eosinophils, leukocyte proteins, and showed that the associations only got stronger whether we looked at biomarkers continuously or as quartiles. Coming to clinical application, the key test that matters is the discrimination, and frequently we use AUCs to understand the accuracy of diagnosis. And here it shows that the biomarkers TNF alpha or IL-9 have a 84% accuracy when used for diagnosis of AIN, and this is compared to 62%, which is like a toss of coin. If you ask the clinician what is their top diagnosis before going into the biopsy, when you compare it with clinical model, once again, the biomarkers outperform the clinical model. Frequently, our top differential in the clinical setting is ATN, and when you compare ATN versus ATI, we see once again these biomarkers do quite well. So if we had to use this either for clinical management or even for drug development, we are trying to separate the, we are trying to have the differential for clinical diagnosis of acute kidney injury. And the two big buckets would be immune mediated, which is AIN, or ATN and other causes. And we can use biomarkers such as TNF alpha or IL-9 for this, or we can use urine NGAL or KIM1 to rule out ATN, but with a urine sample, you can differentiate this, whichever research or clinical setting. And if you end up with high risk for or high suspicion for AIN, you can remove the offending drug or start an alternative agent or use steroids or anti-TNF alpha therapies. And one last slide, which shows that these biomarkers are not only diagnostic, but they have prognostic value. And IL-9 greater than 0.66 demonstrated higher GFR at 6 months in these patients, as well as higher responsiveness to steroids. And this is the p-value showing that high IL-9 has a higher GFR when given steroids versus patients who did not have high IL-9, showing a marker of therapeutic responsiveness. So in summary, AIN is a histological diagnosis characterized by interstitial infiltrate, tubulitis, and eosinophils. We need a high index of suspicion, and these biomarkers may be diagnostic, prognostic, as well as have therapeutic responsiveness potential. This is a very nice example of precision medicine where biomarkers can be deployed in the setting of AKI and AIN. And please reach out to Dennis or I for any questions or collaborations. Thank you.
Thank you very much, Chirag. As we get the slides up, I'd like to introduce our next speaker, and this is Dr. Yerry Albrecht. Dr. Albrecht is the Vice President of Biomarkers and Precision Medicine at Passage Bio, and he's going to be talking to us about qualification of glutamate dehydrogenase and its use in detecting drug-induced liver injury, as well as how we how the collaboration to obtain the data drove this qualification. So with that, Yerry, I'll pass it over to you right now.
Thank you, Nick. I hope everybody can hear me okay. Yes. And in my 10 minutes, I will be presenting on our experience with a collaboration-driven qualification of glutamate dehydrogenase. And my presentation represent the work of the PSTC of CPath, that liver GLDH working group. Okay, let's go to the next slide. So why we actually are looking at GLDH and why we are qualifying it. unmet medical need is basically detection of onset of liver injury in subject with underlying muscle impairments. In this patient population, ALT doesn't work simply because it's not specific to the liver. As you might know, that ALT was actually discovered as a biomarker of myocardial infarction because a large amount of ALT is actually in muscles. So as a leakage marker, if the patient has an underlying muscle disease and muscle decay, that release of ALT from muscle mask or muddied and makes the interpretation of the data in light of liver injury completely unpassable. So in contrast to this, GLDH is liver specific marker and has a assay available, and it's very useful biomarker candidate. That's how we started. And in order to qualify the marker, I just want people to understand that what it's needed to be done. It's a large, significant amount of non-clinical studies and clinical biomarker evaluations just needed to put together to a package that which allow the scientists, the physicians, the drug developers to really understand the advantages or what GLDH does, and also the limitations. That's also both the advantages and the limitations are important. Okay, on the next slide, please. Because it's a large amount of work, and there is no one institution can do it alone. And I, based on my experience since early to mid 2000s, working with PSTC and Cpath as an organization provides the ground for getting the large collaboration between industry, academia, and other research institutions together and have resources to actually push qualification forward. It's absolutely phenomenal. In our case, the alliance, or as I always say, biomarker development takes a village, included industry, pharmaceutical companies. We had 15 pharmaceutical companies contributing a non-clinical data. We were able to evaluate a GLDH and compare it to ALT using large number of model agents in non-clinical studies and use histopathology to characterize it. We had the clinical studies were actually enabled by a collaboration with University of Michigan and Dr. Ken Johnson, where we developed a approach where we are using leftover samples from routine medical exams in prospective way to design which I call phase 0 biomarker evaluation studies. And use those to understand the performance of, in this case, GLDH in populations of patients and healthy subjects across ages, sexes, and also ethnicities, different backgrounds, genetic backgrounds. Another we had from academia provided quite a bit of and valuable expertise, clinical expertise from Ken Johnson or for Paul Watkins from UNC, which help us to move forward. And while I was speaking about the genetic background and different populations, we have a very successful collaboration with NIHS in Japan, where they ran a biomarker evaluation studies, and that allows us to compare a Japanese population versus the Western or African American and so on, and have a large study where we can evaluate an effect of a genetic background on levels of GLDH and allow the qualification also in Japan. So on the next slide, if I may, I'll just kind of give you a glimpse of how long all these things takes. So, and GLDH is a relatively simple biomarker leakage enzyme, like the ALT. Then when I consider the first publication of the qualification seminal paper, which we published in 2013, as a beginning of the qualification, and then consider that we are at 2022, where we are just before submission of a full qualification package. So it's about 10 years of work where we accumulated data over 5,000 subjects, and which are included in all exploratory and confirmatory studies. And as I said, we had to develop in order to enable it with the University of Michigan, with Ken Johnson, the prospective biomarker study design, which enabled to develop the diagnostic criteria and clinical performance of GLDH. We also, all these large data sets includes data from clinical trials from Pfizer, from catabases, where those are clinical trials with Duchenne dystrophy patients, which allow us to actually pressure test or evaluate the performance of the biomarker in clinical trials. And the last part, which is really important in terms of qualification, because when we speak about qualification and people say it takes forever, it's a really long-term process. Does it really, does it really make sense in terms of, does it make a dent on a drug development of individual projects? And to that, I'm saying yes. The qualification process, it's really important for that biomarker to have the to carry the conversation with the regulatory agencies, with scientists at the FDA, EMA, and also PMDA in this case, to share the data, to share the understanding, which is really helpful then to get the biomarker accepted by the regulators under the individual INDs. So yes, GLDH has been used in clinical trials for decision-making under individual INDs. And this is most important to understand, that we don't need to wait until the biomarker is fully qualified, but under IND can be used. And allowed, provided the data and the context of use and the patients allow that in light of what we want to do. All right. So on the last slide, on the next slide, I have a kind of to demonstrate the impact of the GLDH. It's actually in use. So what we did with Nick a couple of months ago, an analysis of clinicaltrial.gov, and search how people are actually using and where GLDH is in clinical trials. And what we have found, besides the project which we know of, a number of clinical trials, mainly from drug development programs, which are for treatment or developing of drugs for Duchenne dystrophies and so on, but also a large number of projects, trials in liver disease. So we even see now uptake of papers after we published our publications from a variety of institutions. So which suggests that all the work which we have done and PSTC really pushed and helped us organize it has the fruits. So at the end, I just want to the take-home message is really biomarkers takes a village, takes people coming together. It requires an institution. They actually put it together, because we all develop biomarkers on as our not a main job. So it's important to have an organization like CPath, like PSTC, who actually can keep us all honest and push forward to get that qualification done. And on my last slide, I'd like to, thanks to Collaborators, there's many people which involve over the years from all the PSTC companies, mainly the data provided by Pfizer, databases, all the people at Cpath, and also the Duchenne Dystrophic Regulatory Consortium. But the people they got involved in generating data and really pushing over the years, Shelley Schumacher, David Potter, and Kim Vanetta from Pfizer, Ken Johnson and Warren Roscoe Warren from U of M are absolutely essential to setting up the collaboration and the design and performing the clinical biomarker studies. Paul Watkins for all his input, and our Japanese friends, Yoshiro Saito at NIEHS, for collaborating with us for over the years on the Japanese and Western populations. And of course, the PSTC leaders, John Michael Sauer, Laura Hopkins, and of course, Nick, to push that all together. Thank you very much for your attention.
Thank you, Yerry. And now I'd like to move on to our next presentation. For this, it will be Dr. Matthias Kretzler. We introduced him yesterday. I'll give a brief introduction once again. Dr. Kretzler is the Warner Lambert Park Davis Professor of Medicine and in Nephrology, as well as a professor in computational medicine and bioinformatics at the University of Michigan. And he'll be talking today on the Kidney Precision Medicine Project, particularly looking at acute kidney injury and tissue that they've collected from biopsies and across sites. So with that, Dr. Kretzler, I will pass it over to you right now.
Thanks so much, Nick, and really thanks a lot for giving us the opportunity to present the Kidney Precision Medicine Project. Many of the lead investigators of the KPMP are on the call. Richard Knight is one of the key leaders in the network as one of the unique features of KPMP as well. The goal of the next 10 minutes really is on one hand to introduce to you what we feel are quite valuable data sets which are publicly available to the community in KPMP already as of today, but also to introduce a framework of developing a data generation, integration, and most importantly, data sharing pipeline. KPMP is a very unique effort initiated by the NIDDK to safely obtain research protocol kidney biopsies from participants in the study who would not need a kidney biopsies. As you have heard, that acute kidney injury and many chronic kidney disease are not routinely biopsied. And our patient volunteers allow us to bring that tissue to the research pipeline in KPMP, where the goal is to redefine these diseases in mechanistic terms, to use that information then in a kidney tissue atlas for broad access of the scientific community to develop new treatment strategies. And as I have mentioned, and Richard already introduced to you, really patient engagement, participant leadership is key in KPMP throughout our process. So this network is set up strategically to have recruitment sites across North America to bring AKI and chronic kidney disease to the study. A central hub is coordinating clinical data and sample acquisition, storage, and sample distribution to leading interrogation sites. We extract the key molecular insights from the tissue in its spatial context and bring that back then to the data sharing instruments. And NIH had the foresight to really give us also a robust opportunity pool where we very dynamically and flexibly can bring the cutting edge research progresses, which we are seeing very rapidly happening in this space. What I really would like to stress is that it really was transformative for all of us to see what key role participants in KPMP can provide to the study and to the community at large, where we see our patient participants provide leadership in many areas, which I have depicted and summarized on that slide. And Richard Knight certainly can give you more insights into panel discussions later, how transformative this really has been for our community. The workflow, as I've included already, is going from patient recruitments at participating sites, including robust biopsy, but also non-invasive biospecimen acquisition, clinical prospective phenotyping, central acquisitions of samples and data, distribution, integration, and sharing. Our patient volunteers are the true heroes of that study because they really donate the risk to donate 3 biopsy cores specifically for a prospectively designed, carefully trained, and audited protocol performed by our recruitment sites, where the tissue is processed using conventional tissue processing technologies to provide the diagnostic insight. Chirag already has mentioned to you, gives in many instances in these not clinically indicated biopsies a lot of clinically actionable findings, because we are indeed finding pathobiology which changes the care of the patients. We generate a second core using a flurry of different OCT, frozen tissue, and then a third core for specialized technologies to be utilized. Here you can see the summary of the technologies we have live against those tissues, all very carefully adjudicated in a rigorous tissue interrogation technology developing protocol against unaffected parts of tumor nephrectomies, and only after careful vetting of the biopsy tissue interrogation sites, these go live against the tissue and then are continuously monitored for performance with very rich metadata captured throughout the process. A key philosophy in KPMP really is that we embrace rigorously the concepts of open science, and data are shared immediately after completion of quality control, not after publication, immediately. And early on, we rigorously identified what are the needs of the communities we are serving. And I think this is probably one of the key take-home messages also for the discussions today, is that really understanding what the needs are. And the needs are, from our stakeholders, very different. Our patient participants have unique interests, and we prioritize those first, and we are serving them to our abilities right now already. And then you can see from pathologists, clinical researchers in academia and industry to professional data mining. We have developed a series of different collaboration opportunities, and I will introduce the key elements to you right now. For, as we discussed yesterday in the breakout sessions, it's absolutely instrumental that we know the experimental context and the generation of the data, the associated experimental protocols, the metadata standard, and the reagents used. And please, here you can see the collections of these different elements which are continuously updated, vetted, and shared. For the data sharing resources, we obviously are sharing the data as they are in Data Atlas repository. And the vast majority of these data are openly accessible to the research world in kidney disease. So if you go to atlaskpmp.org today, you can download all of our data from our AKI patients, except those data sets where we had concern that the clinical data would be so rich that re-identification of study participants would become a possibility, and they are behind the data use agreement firewall, but also heavily used by the scientific community. We then build an Explorer tool as an entry level into the data, how to use them and to access them for the non-bioinformatic expert. And we build finally a spatial viewer with tissue context, molecular information now coming live with the emerging technologies to link the molecular to the tissue context. I want to give you a couple of introductions how these data sharing tools work. The Kidney Tissue Atlas Explorer, we really learned from our user community that the two key entry points are: one, to search via a gene or molecule, and second, to search by a cell line or tissue context. So you can either enter the gene of interest here, or you can deselect your cell types of interest, or even you can just highlight your cell type of interest. And then you are taken to what's uniquely expressed in your cells of interest in the reference states, in the disease states of AKI or CKD, or you can take your gene of interest and you get various displays depending on the technologies used. Here with a single cell data sets, you identify which unique cell type is expressing your cell transcript of interest and which other cells are co-expressed. And all these data are dynamically downloadable for your own consumption with your internal research tools if you would be interested to do so. A very exciting advance, I think, for all of us is the level now that we can take tissue sections of a kidney biopsy and not only captures the structure in a digital format and use advanced machine learning approaches to extract knowledge from the tissue level information, but we can map molecular information on top of those. And we have loaded into the KPMP spatial viewer already the first series of these tissues, where you can select what you're interested in imaging or in patient characteristics. You get a dynamic interface where you can dive into the tissue and define what degree of damage is present in the tissue. We have 6-channel immunofluorescence 3D cytometry, where we can identify the different nephron segments based on their unique characteristics of molecular expressions in their 3D dimensional context in the concept of AKI or CKD, DKD. And the recent advantage of spatial transcriptomics allow us to use specific molecules of interest and then see in the tissue segment where they are enriched in specific molecular compartments, again, contrasting our reference tissue with the AKI and CKD, DKD samples. The Atlas repository is a key instrument if you have bioinformatic firepower available to yourself. We have over 2,800 data sets available there for download and integration in your internal bioinformatic pipelines if you are interested to do so. And as I mentioned, the data are stratified based on technologies, disease states, and also on open or data use agreement protected access. A tool probably most relevant for the discussions in this setting here is that we are now matching the tissue level insight from our patients with a vast panel of non-invasive biomarkers. Many of the speakers here with expertise in their domain are generating not only the acute kidney injury, DIKI biomarkers we have been discussing, but very robust non-invasive biomarker profiles, which are currently undergoing QC, and we hope to share them by the summer with the community at large, so that you can actually download the information on various biomarkers and then can identify what tissue level damage patterns can be linked to your biomarker assays. And conversely, if we can identify and redefine the molecular diseases in the kidney, how do these subgroups relate to your non-invasive biomarkers? I want to point you to a paper which is up in bioarchives, which rich supplemental information available, which is a good example how we are using that information as a community to identify healthy and injured cell states and niches in the human kidney. This is a joint effort of the Kidney Precision Medicine Project with the Human Cell Atlas and the HubMap network, where we profiled over 400,000 cells from 84 tissues, have 102 million different neighborhoods defined in the continuum of reference tissue, AKI and CKD. And yes, you heard me correctly, we consider this based on our molecular data, more continuum of disease trajectories manifesting in the various contexts than binary events. With this, I would like to wrap up. I hope I've given you an impression how KPMP has established a unique community to drive us forward with the insight in acute kidney injury, which really has not been accessible before to date. The critical role patient participants play in that, the critical role of a very lively interactions between NIDDK program staff, the academic community, our patient community, regulators, and partners from industry. And with this, I would like to wrap up and most of all thank our patient participants for really making this study a reality in 2022, and are looking forward to discussions in the panel sessions later. Thanks so much.
Thank you very much, Matthias. All right. Well, I will be giving the next presentation, and I've titled this Advancing Novel Psyche Biomarkers Through Collaborative Endeavors. And so what I wanted to talk about, let's go to the next slide, are within Cpath, we have two safety-related data sharing projects. The first is within the Predictive Safety Testing Consortium. We have what we refer to as our qualification database, and that's across all of our member companies which have submitted data as part of submissions to the regulatory agency for either letters of support or for a full qualification. we have stored that data, and it's something that's available to members to utilize. And I'll get into a little bit of a use case, and then Yemi after me will actually go into how they came up with a very nice publication looking at data that we collected in rats and had stored with just a different question, and was able to come up with some very nice results. And then the other is the biomarker data repository, which you've heard some reference to throughout the past 2 days. And that was a result of looking at what was possible with our PSTC database within the consortium and beginning to apply that to a broader audience and stakeholders. So let's go to the next slide. And so here, in terms of CPath, we when we are handling data and bringing it in, we've built up a very nice staff of experts around in our data collaboration center, who they provide large-scale data solutions for scientific research. We have customized platforms for improved collaboration. They have multi-source data aggregation and standardization, and then as well expert in curating and administration of the data and its storage, as well as having robust security policies as we'd expect, and then application of current regulations to ensure compliance. It hosts a large data set across different diseases and safety data, as well as applies to PSTC. There's more than 200 clinical trials, more than 160 non-clinical studies, and it represents data from more than 180,000 subjects and over 215 million data points. We do this by working across all stakeholders, and we work in conjunction with the Data Collaboration Center within each of our consortia to ensure that the data that comes in is mapped and is useful for its current purpose, and if we have permission, future applications and questions that arise to pursue. Let's go to the next slide. And so, one of for PSTC, one of the things that we have currently two separate databases here. One is our normal healthy volunteer study database. And we've conducted now 2 separate normal healthy volunteer studies, one in the US and one in Japan. And as part of one of our qualifications, we also have some additional healthy volunteer data that was shared from one of our member companies. And then the other is the non-clinical data, which spans several types of several toxicities with kind of your prototypical compounds, as well as some that are compounds that were in development, but for whatever reason were halted. Some of those due to the toxicities that make for very nice models for either liver, kidney, skeletal muscle, vascular injury, testicular toxicity, cardiac hypertrophy, and pancreatic injury. And so this is something that we utilize first for regulatory qualifications and publications as well. And then as well, if we have permission within the database to pursue additional questions to probe the data to see if there are ways that either looking at an additional statistical model may provide additional insight for applying the biomarkers in a different manner. Let's go to the next slide. And so, so how do we do this? The incoming data comes in. We work to harmonize that data into a standard format for our non-clinical data. Each working group, the tissue or organ that is specific to that working group, develops a histology lexicon so that we can apply the same scoring of injury across companies and injuries, and then also harmonize the units that are being used in the biomarker results. That permits summary reports, analysis data sets that we can then share for qualifications and summarizing in publications following that. Something similar is what we are proposing and are going to use in BMDR, the biomarker data repository. And let's go to the next slide. And so, yeah, so for building for the future, as was mentioned yesterday by Amita in her talk around the biomarker data repository, it was a pilot that was launched around 2018, following a few workshops where it was brought up that there is, a large amount of data that may enhance the qualifications that we've already pursued, or perhaps even expand on those qualifications. And that if we were to create a central location to store those, and a central location to store these, as well as curate and standardize so that we could integrate these studies into one database, we could result in additional drug development tools or biomarkers. And so, so far as the pilot project, we went to the Predictive Safety Testing Consortium's members looking for studies that they had either already contributed to PSTC as part of the qualification, or perhaps new data that they have that they would be willing to submit to this database. And so with that, we have received a small number of studies to date, and are working right now to, we've mapped quite a few of those. We created standard formats for non-clinical data to be submitted in templates. And then as well for clinical data, we prefer data coming in the CDISC SDTM format, but also have the capability to map data if it isn't in that format. We also have found that frequently there are slightly different interpretations on the SDTM format. So there's usually a minimum level of curation and mapping that's involved. And so what we're, one of the pieces that we're looking at coming out of this workshop is additional contributions to this repository. And in addition to building the repository, when we launched the pilot, let's go to the next slide. Dr. Janet Woodcock at the FDA at the time wrote a letter encouraging companies that are utilizing these biomarkers in INDs or new drug applications to share the identified data into our biomarker data repository in order to further expand our understanding and potential application of at a minimum the kidney injury or kidney safety biomarkers. Our goal is initially with the kidney safety biomarkers, but also to expand it to other tissues and organs. So now, so just some of the output and access capabilities, we're still defining some of these, and that's one of the actions coming out of this workshop is defining some of the access levels and types of reports. But we have, standard components I already mentioned in terms of summary reports, specific analysis data sets, and then as well establishing the access levels based upon the contributors' comfort level with sharing their data, as well as looking at approved researchers and submissions to health authorities in the future. So let's go to, I think that was my, and so the last slide is, if you are interested in contributing to this, it is, we're looking for anyone with clinical, non-clinical data. Right now, we're working primarily with kidney safety data. And so please do reach out to Katrina. We have a data contribution agreement that is put in place between Cpath and the contributor. And depending upon your contribution and your willingness to share the data with others who contribute, permits access to the data that is in the database. So we're looking at, sharing and contributing data to this permits access to the data at this point for additional analyses. So thank you for listening to this piece, and I look forward to the next presentation, where I think Yemi has a very nice case where there were no new studies, but there were new findings by probing some of the PSTC non-clinical kidney safety biomarkers. And so with that, we can move into the next presentation. And Yemi, I will permit you to give your presentation, but just a quick introduction. Dr. Yemi Adedeji is a principal pathologist in the Department of Safety Assessment at Genentech, where he's been since 2016. And with that, Yemi, I will welcome you and pass it over to you right now.
All right, thank you for the introduction, and thanks to the organizers for giving me the opportunity to talk about, how we're able to use some data within the PSTC database to generate new insight on the use of the urinary biomarkers. Next slide. So there are two things I'll be talking about today. One has to do with finding the optimal urinary biomarker normalization method in non-clinical species. And then the other one has to do with finding the best biomarkers to use for when you're working with mice in drug development studies. Next slide. So for the first one, can I go to the next slide? So PSTC over the years, I've done a lot of work, in qualifying the novel urinary biomarkers in rat for the early detection of renal injury in drug development studies. But one thing we realized in the PSTC nephrotoxicity working group is that users or different companies, they normalize these biomarkers in different ways. We realized that some use urine creatinine, some use urine volume, and some just leave the biomarkers unnormalized. So because of that, it's difficult to compare data from different sources, and nobody really knows what's the best way to normalize this biomarker. So in some ways, some might actually be doing this wrong. So then another thing we realized was that more recently, some people were also proposing using urinary cystatin C as the urinary biomarker normalizer, given some of its characteristics as a glomerular filtration marker. Next slide. So given this, we decided to conduct a study to identify what is the optimal method to normalize this kidney biomarkers for in cases of drug-induced kidney injury. And we believe that if we can figure this out, then we can propose a method that can be used in the field, that the field can adopt and provide uniformity in the field. Next slide. So the steps we took was that we went through the PSTC database and looked through all the renal biomarker studies that have been conducted in rat by member companies. So these are all the data that we have in the PSTC database, data that were provided by the member companies. So I set up some inclusion criteria, which I have listed here. And based on this inclusion criteria, I was able to find a study which was a cisplatin induced kidney injury study that was done in Anwister rat. So overall from this study, they got like 90 samples because there are a total of 90 rats that they used in that study. Then given the fact that we want to make sure that whatever results we generate from this is something that is not just strain specific, it's something that can be used for any strain of rat. We also decide to, we use this criteria that I set up here to find another study where they use Sprague-Dawley rat. But in that study, they don't have as much samples as we had in the Anwister rat. But we felt like that would be a very good control for us to make sure whatever results we generate from the Anwister study is something that can cross over to any strain of rat. So overall, the total amount of samples that we got from this that we worked with was from 110 rats. So this was sizable enough for us to generate a lot of data and to also help us to have confidence in any kind of data we get from this study. Next slide. So the major questions that we asked was, the first one was to understand the variability between all these different normalizing factors that people use. So like I said, some people use urine volume, some use urine creatinine and urine creatinine. And then again, some people are also proposing urine cystatin C. So the first question was to understand the variability between these three factors. The next one was to also understand, when you normalize this biomarkers with any of these normalizing factors, what is the degree of variability of the biomarkers after the normalization? And then the third one was to also understand, do these renal biomarkers still maintain their sensitivity after you normalize them with this biomarker? So the main gist is that we want to know how this normalization method affects the sensitivity of the renal biomarkers. Next slide. So for the first question that we asked in comparing the variability of these different biomarkers, we realized that actually urine creatinine appears to be the less variable among all the different urinary biomarker normalization methods. And as you can see in the first table that I have on the left, overall, either way you look at it, we looked at this either between days or between rats, and we also in males and females. So the male is what I have on the left, and then the females is what I have on the right. So overall, either way you look at it, between days and between rats, urine creatinine appears to be less variable compared to either urine cystatin C and urine volume. Next slide. Then the next one was to understand, to compare the variability of these renal biomarkers after you normalize them with either urine volume, cystatin C, or urine creatinine. And again, maintaining the consistency that we got from the variability result between creatinine and all other factors, we realized that when you normalize these renal biomarkers with urine creatinine, you still have less variability compared to when you normalize with cystatin C or urine volume, or when you leave it unnormalized. And the data that I have here again is either way you look at it, either between days over time or between rats. When you normalize with urine creatinine, you have less variability. So, and this is one thing you want to make sure you have when when you're analyzing your urine biomarkers. You don't want something that's very variable that will be affecting your results. You want to have a consistency when you're generating your data. So this is another proof that urine creatinine is like the best way to normalize these biomarkers. Next slide. And then the final question that we asked, major question that we asked, was to understand, the sensitivity of these biomarkers when you normalize them with these different normalizing factors. And again, from what I'm showing here, overall, urine cystatin C appears to be the worst biomarker that you can use to normalize these biomarkers, because the sensitivity is very, very low when you normalize with urine cystatin C. But the sensitivity is kind of comparable when you normalize with urine volume or urine creatinine. And then when you leave unnormalized, if you leave your biomarker unnormalized, then you also don't have great sensitivity with that too. So next slide. So overall, putting all this data together, it looks like urine creatinine is the less variable when you by itself or when you normalize with the biomarkers. And then the sensitivity is also comparable with urine volume. So putting all those factors together, we believe that urine creatinine is the best biomarker to use when you are to normalize these biomarkers when you are doing the analysis of these biomarkers in rat. So we believe that this study kind of helped fill the gap in the field to ensure that we have an optimal method to normalize these biomarkers, and we believe this can help the field to achieve uniformity in the way we analyze these biomarkers. So this ends the first aspect of the talk. So next slide. So this one, like I said, PSTC, we've been doing a lot of work on characterizing these biomarkers in rat. But one thing we realize is that there's really no much information on, the utility of these biomarkers in mice, which is another preclinical animal species that is widely used in drug development studies. Next slide. So given this, we decided this is one question that we can ask, like, how do these biomarkers perform in non-clinical animal species. Next slide. So the step we took here was that we don't have any of this data in PSTC database, but one thing we did was that we asked our member companies if they have any in-house data that they can share with us that they've used in the past in their companies, if they're willing to share this data anonymously, and to see if there's something we can work with to understand how these biomarkers work in mice. So the three major companies that shared the data with us were Janssen, Merck, and Sanofi. Next slide. So I'm just going to go straight to the result. So regardless of the assay platforms that they used or the nephrotoxic agents that were used or the mouse strains, one thing we realized was that urine albumin and NGAL were like the best urinary biomarkers for detecting drug-induced kidney injury in mice. KIM1 also did well, but regardless of the assay platform, but there was one study where there was no effect in KIM1, which is the table 6 that I have there. And the reason for that was because that compound actually caused distal tubular injury, but you have expression of KIM1 at the proximal tubular injury. So that's the only reason why there was no detection of KIM1 in that study. But in cases where the primary injury was proximal tubular injury, which is what I have in table 2 and table 4, KIM1 also performed well. So overall, next slide. Putting all this result together again, we were able to show that in mice, urine albumin and NGAL are like the most sensitive biomarkers when you're trying to detect drug-induced kidney injury in mice. So, and then KIM1 also is a very, very good biomarker in mice as long as the injury is limited to the proximal tubular injury. Last slide, please. So overall, I think that we've been very, very lucky with PSTC and our member companies that they've been willing to share all the data that they generated over time, either in-house or the ones that we have in PSTC database. And this data, they've enabled us to gain more insight into the utility of these biomarkers in non-clinical animal species. And I think that's the last slide. Thank you.
All right, thank you very much, Yemi. We appreciate that, your presentation and your dedication to PSTCs, advancing the data we've collected and reusing that data for additional questions. All right, well, we are going to move into our panel discussion right now. As a reminder to our audience, you may ask questions of the panelists here in the chat feature, and we do have people monitoring this. I will ask that our presenters from this first session do put yourself back on video, and we will move into our first set of questions. All right. Let's see and so, yes, as I was mentioning, you can please put your questions into the chat if you have them. And so I think, first, Chirag, perhaps, you can kind of expand. What do you see in terms of looking at the AIN biomarkers? What do you see as the next steps for implementing or looking towards broader acceptance? What types of studies do you see as being needed, and what actions do you foresee?
Absolutely. This is a great question, and we have been thinking about this and doing studies along those lines. So there is the academic collaboration for identifying immune-mediated injury is growing. So there are 4 centers that are participating in kidney biopsy studies where we collect blood and urine samples and validate these results. It would be ideal if we can test these biomarkers in completed clinical trials where if there were certain agents that were thought to have this side effect, and whether we could have picked it up early, that would be a very nice proof of concept. We are also trying to do the studies in KPMP. Dennis has ancillary studies submitted where we will get better tissue-related pathways that are going along these biomarkers to corroborate the evidence. And we can optimize the assays. We can do point-of-care systems so that we can use it clinically. This is a type of marker where we would like to have the results available relatively quickly. It's not the chronic setting, so we can stop the drug, etc. So developing those aspects and having industry collaboration would also be ideal. But the promise is huge, where with one urine sample, you can identify injury, NGAL and other markers. We can identify immune-related injury and maybe other biomarkers if they're out there.
So, and Yemi, for, in your studies, in the normalization that you've used, do you see this applicable to other species?
Yes, I think for other non-clinical animal species, I think yes, because most of these animals, they are exposed to the same environmental factors, unlike in humans, where humans are exposed to different environmental factors, and that could affect the amount of creatinine that you have in your muscle mass. In the animal studies we used, most of them, all those animals, they have the same exposure to the same environmental factors, and because of that, I think that makes urine creatinine a very, very good method, that we can use not just in rats, but even in mice and in non-human primates. I think this is something that can cross over within all the non-clinical animal species that we use for drug development studies.
And we do actually, there's a follow-up question from the audience for you, Yemi. And I'll just read this question off to you. Since urine creatinine drops in AKI, at least until a steady state is reached, it's expected that dividing or normalizing to urine creatinine would make a biomarker that increases in AKI appear more sensitive. How would normalizing a biomarker that normally decreases in AKI, for example, Uromodulin, affect their sensitivity?
So can you go over the question again?
Yeah. So, and so the question, the meaning of the question is, how would normalizing a biomarker that normally decreases in AKI affect its sensitivity?
Oh, okay. So it actually increased the sensitivity because we showed the result here. The last figure that I showed was actually in cisplatin-induced kidney injury, where we truly have acute kidney injury. And the sensitivity actually increased when we normalized with urine creatinine and with urine volume. Their sensitivity were comparable between those two normalizing factors. So with DIKI, it increases the sensitivity.
Thanks. And a question to the whole panel. And I'll look for most of you for a short comment, because we're just going to take 15 minutes for this this session, but we have a few extra minutes. The panel comment on how academia and industry can better collaborate to advance the development of promising biomarkers of drug-induced kidney injury.
Nick, I have a very short answer to this.
Okay.
The very short answer to this, please, whoever is interested, give Nick a call and work with PSTC.
Well, thank you very much for that, Yerry. Matthias, with KPMP, how do you see engagement between academia and industry in the long term? Oh, I think you're on mute right now, Matthias.
Okay, can you hear me now?
Yes.
Okay, so I think it's happening already. Our data are openly accessible and are heavily utilized by the entire biomedical research community in kidney, particularly pharma. In addition, we have data use agreement mechanisms in place if rich data sets from the clinical space is of interest, and that is heavily utilized by the private sector as well. In addition, the samples we have procured, as you have seen, we have generated comprehensive biomarker profiles, both with targeted assays run by experts, many of them on this call, or by large screening platforms. And again, they will be openly accessible. If there are unique assays and needs of interest to companies, then they can come to KPMP for auxiliary study to access those samples, even access the patient populations, after obviously appropriate auxiliary study review by the group. There is another step which I hope we could see happening in the current starting funding cycle of KPMP, that as companies are engaging into clinical trial activities, to what extent samples and activities from clinical trials could be interacting. And this can be a bidirectional effort, so that we have all of our protocols in the public domain, and several of the company efforts associated with clinical trials are already utilizing the KPMP protocols for their activities, so the data can be harmonized, or conversely, samples data sets procured by companies can be brought forward. The final step is the data sharing instruments are key here. And as we have been discussing, finding creative ways to share data from private entities, I think, is critical, and submitting them to a central data repository like BMDR obviously is the ideal scenario, but there are now even scenarios available with federated databases that data can continue to reside by a private entity, but can be centrally accessed, analyzed in a protected manner, and then aggregated data exported out. I think there are many different ways with the innovative tools and studies in place that we can push our agenda forward together.
And Chirag, how about with the work that you've been conducting right now?
In terms of collaboration with industry, or?
Mm-hmm. Yeah.
Yeah, so we have assisted the trial design or the protocol development of certain drugs. So there are at least 2 examples where hemodynamic injury was suspected with drugs that were being developed for liver disease. And we were able to rescue the phase 2 studies because we measured a complement of biomarkers demonstrating the creatinine rise was not associated with increase in biomarkers. We have also taken our data and combined it with a lot of other clinical trials and identified some biomarkers that could be used for prognostic potential or pharmacodynamic setting in CKD trials. So we are open to a lot of collaborations in various ways, intellectual data, or even actually measuring samples when needed. I would think that if this pipeline became much more open, that every trial, if samples were collected and put aside, even in the post-hoc setting, certain biomarkers could be tested once the trial is completed when better idea is available. I know this is expensive, but as a community, we will go much farther in terms of knowledge generated from each trial.
Well, it looks like, Yemi, we have another question for you. And so one is, do you have data using total urine protein to normalize the biomarkers?
No, we did not use total protein. I would like a follow-up from the person to know the reason why they asked for total protein, because yeah, there will be a lot of variability, I think, using total protein, depending on the kind of injury you have. So yeah, that's not something we used. cystatin C it's a kind of protein, and we saw how that panned out. It's a good glomerular filtration marker based on some of its characteristics, and it was like the worst. So that's one example of a protein in the urine that I can say that we used. And yeah, in terms of the sensitivity, it wasn't good. And then in terms of the variability, it was very, very variable compared to creatinine and urine volume. So if I'm actually going to, categorize, how this, how they stack, I would say creatinine is the best, followed by urine volume. And then, in fact, leaving the biomarker unnormalized was actually better than normalizing with cystatin C. So I will assume that normalizing with total protein might also be a problem.
And one additional question, what is, that your data suggests that urine creatinine and urine albumin provide the most valid normalizers and biomarkers in rat and mice mean so what does this mean for the more novel biomarkers? How do you assess whether the new biomarkers beat urinary albumin in performance?
So I hope the person is not mixing like those two talks. So the second one was like trying to understand, the performance of those biomarkers in mice. And what I said then was that urine albumin was the best, along with NGAL, regardless of the assay platforms that were used, or the rat strain, the mice strain, and regardless of the nephrotoxic agent. Urine albumin was very, very good in detecting proximal tubular injury in mice, along with NGAL. So that's where I mentioned urine albumin, but nothing like that in the normalization project. Or can you go over the question again?
Yes, let's see. So the question is, what does the fact that Dr. Adedeji's data suggests that urine creatinine and urine albumin provide the most valid normalizers?
Oh, no. So it's urine creatinine that's like the best normalizer.
Yeah, urine creatinine.
Yes. And again, when we normalize with all the different biomarkers with albumin or any other thing, all those biomarkers maintain their sensitivity when they were normalized with urine creatinine, either in the presence of tubular injury or without any tubular injury. So, and albumin was one of the markers that we used, that were available, that we use in detecting drug-induced kidney injury in our non-clinical animal species. So albumin performed very, very well, and when it was normalized to urine creatinine, it still maintained its sensitivity compared to urine cystatin C, or if you leave it unnormalized. Oh, what does the fact that suggest that? Provide. So yeah, I'm saying the question again. So urine creatinine, yes, is the best normalizers, whether in rat or mice. And urine albumin is one of the best urinary biomarker in mice for detecting drug-induced kidney injury. I think that should clear things up.
Thank you. And Yemi, I think that you have a question for Chirag, if you don't mind asking that question.
Oh, yes. So my question is, from your presentation, you said that most patients are not symptomatic, with AIN. So my point is, so it looks like when it's detected, it's like the disease might have progressed, So are people thinking of screening methods, like we use, for like cancer patients, for someone that already has cancer but are not symptomatic? Are there screening methods that you guys are thinking of, that can be, incorporated into preventive care?
No, this is
especially for especially for patients that might be at risk? And what are the factors that you guys have to identify patients that might be at risk that their primary clinician might suggest that they take those kind of tests routinely to screen them for AIN?
Correct. No, this is a great question. There are over 100 drugs that are associated with AIN, and some common agents like PPI, although the absolute risk is low, since they are used in millions of people, there are a lot of patients who have underlying progression because of PPI. So we are trying to address this in two ways. We are trying to get samples from large cohorts such as Craig or Eric, where samples have been collected on an annual basis, and patients who have unexplained progression, and if they have elevation in these biomarkers, we know that there's an immune-mediated injury. And that will give us an idea that using these biomarkers for screening purposes may be beneficial. So next time somebody is on immune checkpoint inhibitor or on a PPI or on NSAIDs for a longer time for the arthritis, should we be checking this intermittently with worsening, say, proteinuria or worsening creatinine, would save a lot of kidneys. And I think that would be a very good use for these biomarkers. I also wanted to round up on the questions somebody was asking, Yemi, that what happens with UMOD and adjustment. Actually, using samples from ASSESS AKI study, which has so many biomarkers, we compared urine osmolality, urine creatinine, and indexing versus adjusting methods. And UMOD is one biomarker that behaves very differently compared to other biomarkers when you index with creatinine versus adjust with creatinine. And you have to be very careful because the levels of the biomarkers go down with AKI, and it becomes a math issue of denominator versus creatinine, which is also decreasing. And the paper goes through this detailed analysis. So each biomarker has to be handled very separately, thinking about its kinetics in the setting of AKI.
Sure. All right. Well, I think we have time for one more question, and this one comes from the audience. There's a lot of discussion regarding interest in collaborating and sharing. Is this an indication that the stakeholders involved so far would like to come together after this workshop and move forward in either forming a consortium or forming a collaboration within an existing consortium to further the biomarker data repository? And I'll open that to anyone who'd like to reply on this panel.
I think, Nick, you might be able to be the one who is most qualified to answer that question.
Well, I know the answer. I know the answer across the space that I would appreciate, and I definitely will be working to advance this piece over the next few months. And so there are lots of people working across the space right now, and there are, I think, related but disparate projects that we have the opportunity to harmonize some of the outcomes, at least as a starting point in assessing drug-induced kidney injury.
Yeah, and I think it's also, as we have seen throughout the last day, as you said, there are many convergent efforts coming into play where with the concept of open science taking root here, allowing us not only to load more data in BMDR and then leverage that for ongoing efforts, but also to use efforts like the one I presented this morning which KPMP has been pushing forward to mechanistically understand the underpinning. And I think that really has been, at least in the AKI, but to my knowledge also the DIKI field, one of the main limitations. This is a syndrome, this is not a disease. And if we can pinpoint with specific markers to underlying pathobiology, then that really can rescue drug development programs, which otherwise would be in very hot water if we understand why, which cell types in the kidney respond to compounds the way they do.
Yeah, definitely having a strong mechanistic understanding is going to be important, and not just treating symptoms.
And Nick, it's Gary. Just one thing in response to Matthias. So one of the clear messages here is, after this workshop, how soon can we begin this process? And I think the answer is very, 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 very quickly thereafter. There can be very little daylight between the end of today's session and the actual sharing. And I'll leave it at that because I know there are other speakers that are coming today.
Thanks, Gary. Okay. All right. Well, I'd like to welcome everyone back to the workshop. And we have a presentation now from Amanda Borens, who is the Executive Director of Cpath's Data Collaboration and Data Science here at Cpath. We look forward to her talk on overcoming barriers to data sharing, some of the why and how that have been successful in other endeavors at Cpath. So with that, Amanda, I will pass it over to you to walk through your presentation.
Great, thanks, Nick. So yeah, I think Nick did a great job on the previous slide. Why don't we move to the next one? Hi, everybody. I'm just going to take a moment to talk a little bit about myself because I don't think I'm very familiar to this particular working group. My name's Amanda Borens, as Nick said. I've been in healthcare informatics or data science for, gosh, more than 25 years, 27 maybe. I am the executive director of the data science team within the Data Collaboration Center. And the Data Collaboration Center has existed since long before I joined Cpath. It's really one of the foundational parts of what makes Cpath a really great neutral convening organization working with consortia in the pre-competitive space. So as the leader of about 10 data science staff, which are part of a much larger organization called the Data Collaboration Center. We work together with data managers, with data privacy officer, with also DevOps groups and operations team. And so really everything that we do is try to enable the different tools and learning work streams from all of our different therapeutic area consortia. I think we're up to about 20, although not all of them are actively sharing data right now. So maybe closer to 16 when we have active data projects happening. And so I just wanted to provide that little bit of backup, sort of foundational discussion about where I'm coming from before I start talking about sharing data, how and why. So we can go to the next slide, please. So let's break it down. Let's start with just focusing on why should we share data? So on the next slide, I've started with a big constituency on this particular workshop, which is for industry. So I think it's really important to talk about the reasons for sharing data from this perspective of the multiple stakeholders involved. So if we talk about sharing data for industry, there were some really great points made in day one of this workshop yesterday about sharing data to identify drug-induced kidney injury biomarkers and tools. So, this is not the extensive list of the reasons, but I think these are really hitting the important points, which is that if you were to share data in order to, as a group, identify drug-induced kidney injury biomarkers and tools, novel ones, obviously, or improved ones, some tools that have better precision that help you to, not exclude too many patients, but to really early on identify the potential for kidney injury so that that could be either avoided or interventions could happen sooner to prevent much more serious kidney injury. All of those things coming from data sharing, the tools and biomarkers that help you to do that, they improve your study design. They help you to avoid unnecessary risk for your patients and for your company. They also protect the study participants, right? But under the category of improved decision-making before and after trials, I think this idea of data sharing that helps you to have better precision tools and biomarkers, it also would help you to enrich for the right patients in trials. It might help you expand your current qualification or context of use. So in summary, I mean, all of the reasons that were discussed yesterday about why for industry are in summary to save money and to save resources so that you can move more quickly and spend less money on therapeutics that may not make it through safety trials, right? So data sharing, in my opinion, because I work across all these different consortia and I see all these different points of view, data sharing has no better cost benefit ratio than this particular paradigm, in my opinion. And that's because in this particular case, you're not sharing data that might help your competitors outcompete. They might, that might give them some sort of insights into a particular therapeutic. You're talking about sharing data for safety biomarkers, right? And so the idea that each of you would individually try to identify and qualify particular biomarkers or tools, such as, progression for kidney injury, for example. The idea that each of you would do that and try to reinvent the wheel from a small pool of the only data that you have access to, which is your own, is just not, it's really not. It's not only beneficial to share data, but it's not really even possible in some cases to be able to do this work without sharing data. So in the next slide, I'm going to switch focus a bit, talk just a bit about why share data for patients, right? You're going to notice that these are broadly many of the same reasons, right? You want to avoid unnecessary risk for patients. You want to protect patients as study participants. You want to improve decision-making before and after trials. And that has the benefit for patients of, saving them the trouble and the hassle of not participating in a study that is not right for them. It also might help to expand your qualification or context of use, which again, helps patients because they might have access to a therapeutic that is really effective for them that wasn't otherwise available. So in summary, for patients, sharing data has many of the same justifications and then some, right? So let's go to the next slide and we'll talk about why sharing data, just my personal experience. And so I am a person with a chronic condition. I see my team of doctors in a research facility, a research hospital affiliated with our local medical school. And so because of the fact that I take medication that has these risks associated, I have to be monitored, and I see them probably more often than most people see their doctor. And for that reason, they know me as this person who, as a data scientist and a biologist, a scientist, I know how hard it is to recruit patients for studies. So if there is a study in the medical school that I can help with that, involves little risk for me, I'm always available. And recently, I participated in a COVID vaccine study, and this was early on in developing the vaccine. It was very initially released to people under certain conditions. And because the medication that I take could theoretically make me immunocompromised, they wanted to build a cohort of patients with my condition and on the same medications to talk about how it might be less effective, and if so, would boosters help, right? So there was this several months of regular visits to look at immunology tests and things like that. And so it's not the first study. I've given tissue, I've given blood, I've given time, I've given lots of things over the years. I think that because I'm both a patient and a data scientist who works in this particular field, You know, it's funny to me, and ironic, but every time I participate, I am shocked at how little data is being shared. And so I remember when my study coordinator was bringing separate tubes to draw the same blood, extra blood from me to do the same tests that were already being done just as part of my routine medical care because they couldn't share the results. You know, it's frustrating to me as a patient because I'm having to give these extra tubes. It just seems excessive. I know that there's a technology solution to this. I know that sharing data would save a lot of the time and trouble and extra visits that I have in order to participate in the study. So, you know, it's cost me a lot of time and pain, honestly, from extra blood draws. I'm one of those tough sticks. Stress, time away from my work, time away from my family, and I'll absolutely do it again if I can help the science. But I really expect you all to share. I expect my doctors to share. You know, I'm constantly evangelizing about that. So I want to talk a little bit about the ethics of sharing, because one of the things that I hear as a data science executive director at Cpath, one of the things that we hear in the DCC is this concept that why should I share these data? Because I have put my blood, sweat, and tears into collecting these data. They're mine. Why should I help someone else's career? Why should I help a competing company's program? And I want to just really take a moment to emphasize that the ethics of data sharing, really the precedent is, and I think there's very broad agreement among medical ethicists that the data belong to the patient. They don't belong to the collector of the data, right? These patients, they're joining in the studies, and I share many of these perspectives. We're hoping to advance the science, right? As a scientist, I'm hoping to be part of that incredibly noble process of collecting information and learning about human biology and medicine and advancing the science that will have effects long after I'm gone. In some cases, patients are joining a study or a trial in this particular example to receive newer and better treatments. But always, I think patients are hoping that a side effect will be that they help future patients. And so, you know, I know of patients and studies that have joined specifically because they know that there's you know, nothing that will change for them particularly, but they really hope that in the future, maybe for their offspring or grandchildren who have a familial condition, that they will have access to better treatments. So these are all of the motivations that I think patients have in joining studies and taking their time and energy. But I don't think that any patient ever thinks, man, I'm doing this specifically to help this pharmaceutical company be better than all the other pharmaceutical companies. So I'm being a little bit edgy here in saying it, but you know, I think it's important to say it out loud. I've never joined a study. I do love my doctor. I hope it helps his career. He was an author on this paper in Nature Medicine. But that is not my motivation. I don't think that's probably ever a reason that a patient joins a study, right? And so I just want to draw attention to that because it's so important as a reason for why we share data. So on our next slide, I'll get down off my soapbox, you guys, and talk about the impact of sharing data. So, you know, how does sharing data have an impact, not just on the field, but on patients? And I think one of the things the FDA have really encouraged us to do is think about the patient experience, the voice of the patient, right? And so sharing data, when a patient is one of 100 or a few dozen, sharing data amplifies the voice and the experience of each of those patients, right? It gives you combined power of patients from multiple trials and multiple locations. And that helps to make sure that that patient experience is represented. It helps people with special conditions or comorbidities that may not be part of trials. For example, when we provide exclusions and enrich for certain types of people, we're wanting to de-risk the incredibly expensive process of getting a drug to the market. And so often certain special conditions and comorbidities are excluded. So sharing data, that's the kind of impact that it can have, is that it provides you with justification and modeling tools that can say actually that based on the results of this trial, people with these comorbidities we could consider doing a study, or in some cases, modeling information might be enough to expand the context of use. So companies all benefit from shared tools that are derived from shared data, right? It makes no sense for the company with the largest research budget to be the one always identifying and getting qualification for novel biomarkers. That's particularly challenging, but it's a place where just sharing data on safety for kidney injury, for example, just sharing data about that is something that would benefit everyone that is involved in the data sharing. Okay, so on the next slide, I want to talk about, Nick asked me to give a couple of success stories. And in this particular case, I'm pulling three. There are lots. There are lots, not just from my career, but from the times at Cpath before me. These are three that I was at least peripherally involved in, so I feel more qualified to talk about them. But the kinds of success stories that I've seen that I think are impactful are reflected in these three. So bronchopulmonary dysplasia is a condition that affects neonates, and because they are neonates and not often studied, there are particular challenges with not just neonates, pediatric populations in general, not often being included in clinical trials, right, and the dangers associated with that. Well, some of the problems associated with always being excluded from trials are also that, excuse me, we just don't know as much about things like normal responses to various medication. So in the case of BPD for neonates, we have a sharing initiative going on that is reaching hospitals, children's hospitals specifically around the US and the UK and Japan. We're sharing data across these different neonatal populations. And we're hoping to identify quote unquote normal lab value ranges for a variety of lab values that might help us to identify BPD earlier and build, at least get closer to building a natural history, or disease progression model for BPD patients. And all of these things are happening with real world data, but they wouldn't be possible without sharing, because there aren't enough patients with enough lab tests, from a single hospital, especially because the population of patients which are neonates with this condition is quite small, right? So it requires data sharing. And I think that's a real success. That's an example of something that we can do that's made possible only by sharing data. TB Reflect is the second one. And if you don't know about it, I've included a link here to an FDA page. And it talks about studying phase 3 failed clinical trials and how we were able to identify subsets of patients and really develop more optimization and precision for selecting regimens of existing drugs, right? So being able to combine drugs in novel ways and to identify some of the things that make patients respond differently. And in that way, we are able to develop a more precise regimen of drugs for individual patients that may have been unresponsive in the past. And the thing that I love about that story is that this was getting companies to share embarrassing versions of failed trials for phase 3. And it was incredibly successful in being able to identify patterns and regimens and PK/PD modeling that are actually having an effect for all companies, right? They're learning a lot about what works and what doesn't work based on the results of all of their failed trials together. The third one I'm going to touch on really briefly because it's so commonly being discussed by Cpath, and that's the RDCA-DAP, which is the Rare Disease Cures Accelerator Data and Analytics Platform. And in this particular case, it's focused on patients with rare conditions, and we're aggregating data, which is particularly important when there's a dearth of data. So there are 7,000 or 8,000 rare diseases. Many of those have never been studied. Some of them have been studied, but have never had any sort of trial for a therapeutic. And what we're hoping in the aggregation and what is happening so far is that we're able to combine data from diseases that have certain things in common, for example, symptoms in common, where a disease that's more studied, even though it's rare, people have heard of muscular dystrophy, for example. So by looking at neuromuscular diseases, sometimes you're able to see therapies that help with particular side effects that are important to the patient that might be helpful in a patient with a disease that is similar but has not been studied. So there are lots of things happening. There's a lot of activity and research going on in RDCA-DAP, and I think those are 3 great examples of success stories. So on the next slide, I'm going to start to kind of burn through my incredible examples of why share data, because it's good for everyone. In summary, everyone wins when we share data. So on the next slide, I'm going to kind of branch over to how can we share data? And I'll start on the next slide with talking about sort of the technical needs for sharing data, and just kind of make a plug for the DCC. So the Data Collaboration Center has so many examples of challenges and concerns that have been raised for, reasons that data sharing is impossible. And we've been collecting all of those reasons over the years, right? And so I just want to talk a bit about why Cpath is particularly qualified to be a data steward, honestly, and to host data from multiple sources and to share them in a secure and private way. Not just a way that protects patient privacy and security, but also that protects the data custodians who are submitting the data. So companies, for example, or academic study owners. In both cases, you can define the conditions for sharing data with our data use agreement. We respect that. It's incredibly important to the DCC that we always respect that. So we're able to sort of meet people at their comfort level, and always we encourage as much open sharing as possible, but we make the data use agreement is a tool that we use to make sure that everybody can overcome whatever their sharing and privacy concerns are. We have currently are hosting, and I actually think, Nick, that this is a bit out of date because I think the website hasn't been updated since 2020. So we've grown leaps and bounds since then. But at that time, we were hosting data from over 80 countries, representing nearly 200 clinical trials for more than 150,000 subjects. I know that it's expanded. We just got the largest data set in the history of Cpath by orders of magnitude just about a month ago, that included patients in electronic health records, right? So in addition to the fact that we have all this experience with security and privacy and sharing data, we also are deeply embedded in the data standards community and have written more than 40 therapeutic area user guides for CDISC. So I've listed some links here in the slide for just references, FAQs. I'm going to run through some of the major things, but I will tell you in summary, there's no excuse we haven't heard. I think this is one that I've heard our Chief Science Officer, Klaus, and I've heard our Chief Technology Officer, Rick, both say this really repeatedly. There's always a conversation about why a particular data owner can't share data. And it's always the 800th time that we've heard that excuse, right? So if you're willing to be collaborative and work with us to address your concerns, we really can overcome any sort of challenge with privacy or concern about security. On the next slide, I'll dive into a couple of examples of how we've overcome some of these. So concerns about legal consent. So if a particular study consent form, if it is not specifically prohibiting secondary research use, there is lots of legal precedent that allows secondary research use. So for example, I'll give you an example. The consent form that I signed in this latest clinical study that I was a part of for the COVID vaccine, it does not specifically prohibit the secondary use. It doesn't specifically prohibit anything, right? I mean, it can be used for research purposes. And my own personal spirit as a patient was that I hope it gets used in any way that it could possibly be helpful, right? So that one comes up quite a bit. It is very rare that a study consent form explicitly prohibits secondary research use. Very rare. In that case, that is also, those are data that are not shareable, but we very rarely have run into that over the last 15, 17 years, something like that. I want to talk just a minute about de-identification of data and how it protects patient privacy. So we have lots of experience. We have legal law firms that we consult with. We have a data privacy officer who specifically focuses on this. And then we have a legal and contacts group within Cpath who have experience within the pharmaceutical companies, many years of experience working in legal for pharmaceutical companies. So we're quite careful and concerned with complying with GDPR and HIPAA. And one of the things that I think also is important to point out is that it protects patient privacy, but it also can protect company privacy and IP, right? So when we're talking specifically about safety biomarkers and sharing data to identify safety biomarkers, I think there should be logically a concern that sharing data might expose the company to risk by sharing, safety issues that they've had in studies. And I just want to allay those fears because we can do all kinds of things to de-identify the data, but also to anonymize it to the trial or the study or the company or even the therapeutic that was involved. We do lots of things like masking out study names. We don't reveal company names. We can combine data in a way so that we have, for example, a recent database that I was working with included close to 30 clinical trials, but they were aggregated in such a way that you couldn't tell which patient record was from which trial. You couldn't possibly re-identify where the particular patient, which study they were in or for what company. So I want to make sure that that's clear to this group. And then on the next slide, being mindful of time, I want to talk about, on the next slide. Great, thanks. I just want to talk about, kind of close on a technical and high note, which is that if we've convinced you to share data and you have had some of your concerns or fears addressed, I want to talk about how we actually do that, right? So how do we share data at Cpath? And I think the reason that that's important is that if you trust us to be your neutral convener and your data steward, I want you to understand how much effort we put into really the science of data curation management and data science in general. So we adopt FAIR data principles. Many of you will have heard of this. Sometimes if you're funded to do research, you'll see that grant funders are often funding on the condition that you will adopt FAIR principles and share your data. So on the next slide, I'll talk about findability. So this is my great example. Some of you of the right age will recognize the Indiana Jones warehouse where the Ark of the Covenant is being lost forever to history. So what we don't do at Cpath is put data into a massive store where we can't find it again. And we can't make use of it because it's just in a box that can't be found, right? Among so many data sets that we can't possibly keep track. So we make data findable with really seriously focus on metadata and case report forms and interviews with the data owners who submit the data to make sure that we clarify semantic meanings of various variables. So findability is something that we're heavily focused upon. On the next slide, I'll talk about accessibility. So Cpath's really pioneered this. I have worked with and been lucky to work with data platforms around the globe. And one of the things that I find the most painful in most situations is that oftentimes, data platforms will profess to have hundreds of data sets. And the absolute truth is that they have access to hundreds of data sets, but when you make a request for data, oftentimes they're going to make a bespoke subset of data that has been de-identified and only includes the certain variables that they think that you need in order to execute on your analysis plan, right? So with Cpath, I think that a way that Cpath pioneered long before I arrived is by identifying this idea, this process of pre-negotiating access to defined data sets. And that means that we get through this whole process of signing a legal document called a data collaboration agreement. And that means that when we've curated and managed data and we've uploaded it to our platform, a person who requests it, who meets the requirements that, again, were set out by the data owner, a person who meets those requirements can access the data as soon as the request criteria are met. So all of these data are accessible and reliable and securely stored in a cloud, either on AWS or Microsoft Azure. And on my next slide, I'm going to talk about interoperability. And I'm just going to touch on it briefly, that metadata and semantics really maximize reuse and interoperability. This is a particularly important concern when you're talking about data sharing, because you're wanting to aggregate data, right? You're not wanting to use one study. If one study was enough, then you didn't need to work with others and share data. So we focus heavily on interoperability. We're focusing on tools and semantics and even the use of ontologies in the DCC and data science team to help aggregate data in novel ways. We also do a lot of work aggregating data across platforms when data can't move. So we're working on developing APIs and federated querying. On the next tab and final one, I want to talk about reusability, and that's particularly a concern as well for sharing data, because if you're talking about one study, this becomes a little bit less important. But when you're wanting to maximize the aggregation of data for a question that wasn't originally intended in the study design, right? A study might have been performed to ask question A, but maybe your question is an unrelated question B. So reusability becomes very important in that scenario. When you're talking about secondary scientific use, reusability is important. And that's why metadata, standardized, cleaned data, contact with data owner to save records where possible. We're really maximizing the value of data. We've been refining our curation process and enhancing this process specifically to enhance the value of data for secondary scientific purposes for more than 15 years. And I'll close by just saying that we really owe it to the patients to maximize the value of their contributed data, not by using it once, but by using it whenever it's scientifically valuable. And I think that we at Cpath take that commitment seriously, that we take that responsibility seriously, that it's also up to us to make sure that when the data are being submitted to us for secondary scientific purposes, that we help it achieve its highest and best value. So with that, I'm going to turn it over to our next speaker, Nick.
Great. Thank you very much, Matt.
Yeah.
All right. Our next speaker is Christine Garnett. And Christine joins us from the FDA, where she's a clinical reviewer and team leader in the division of cardiology and nephrology. She leads CDER's interdisciplinary review team for cardiac safety studies as well. And she'll be talking around data sharing for solutions with her perspective from the FDA. And so with that, Christine, excuse me, pass it over to you right now.
Great. Now, will you be going through control of my slides?
No, just let Katrina know when to advance the slides.
Okay, all right, great. So thank you so much, Nick, for the introduction. And I'm really happy to be here. And I'll be giving a perspective that how data sharing activities occurred in the cardiac space. So I've been working in cardiac safety for over 15 years. And I would like to share with you my experiences, both with what worked and what did not work with data sharing in cardiac safety. And I'm hoping that some of the lessons learned that I will be discussing with you really could help advance the data sharing initiative to increase knowledge of safety biomarkers, improve study design, and tool development for early detection of drug-induced kidney injury. Next slide, please. So a little background on cardiac safety. So mainly it's going to be focused on QT interval prolongation. So prolongation of the QT interval on the surface electrocardiogram has long been known to be a marker of antiarrhythmic mechanism. However, excessive prolongation of the QT interval in the right setting can also trigger cardiac arrhythmias. And these cardiac arrhythmias that we're worried about are called torsades de pointe. Now, beginning in the late 1980s, marketed non-cardiac drugs were found to induce these cardiac arrhythmias, and sometimes with fatal outcome. And this really important cardiac safety risk was not detected during the clinical development of these drugs. So overall, there's been about 13 drugs that have been removed worldwide from the worldwide market because of drug-induced QT prolongation and torsades. And in the early 2000s, this was the leading cause for drugs to be removed from the US market. Next slide, please. So this global safety issue really provoked a comprehensive response from drug regulators, drug developers, and academia, which really resulted ultimately in a decreased number of new drugs that cause excessive QT prolongation and torsade. And this slide just shows the timeline of the response to this unfolding safety issue of drug-induced QT prolongation and cardiac arrhythmias. And I just like to highlight some key parts of this global response. So starting in the late 1990s, regulators began working on guidance documents for both non-clinical and clinical testing for drug-induced QT prolongation, which culminated into 2 ICH guidelines in 2005. One of the guidelines was S7B, which pretty much gave recommendations on non-clinical testing, and E14, which provided recommendations on clinical testing. And today, pretty much every new drug that's in development will undergo some type of both non-clinical and clinical testing for its ability to prolong the QT interval. Now, in the early 2000s, stakeholders generated a standardized data format for digital ECGs. This effort was really, I would say, it was led by the Health Level 7 Information Technology, which is a global authority on standards for health information. And it's really important that for any type of data sharing initiatives, a standardized format is really key for a success. And also in the early 2000s, FDA initiated a cooperative research development agreement, or a CRADA, with Murtara Instruments to build an ECG warehouse based on the standardized format of digital ECGs. And then in 2007, the FDA and Duke Clinical Research Institute established the Cardiac Safety Research Consortium. And one of its core objectives was data sharing of electronic electrocardiograms to develop new tools and biomarkers for drug and device development. So next slide, please. So for the rest of my presentation, what I'd like to do is tell a tale of 2 data sharing efforts. And then I will share lessons learned from our experiences with data sharing. So the first is the FDA ECG warehouse. This warehouse was developed through the CRADA between FDA and Murtara Instruments. And the main purpose was to share digital ECGs with regulatory agencies to support regulatory review of clinical QT studies. And Murtara Instruments stored all the digital ECGs until 2021, when it was transferred to the FDA. Now, the second database is the one through the CSRC. And this database was developed as part of a memo of understanding between FDA and Duke Clinical Research Institute. And the purpose was to share digital ECGs with stakeholders to promote research activities for cardiac safety. And this was a voluntary database, meaning that companies who own the data would have to agree to release their digital ECGs from their clinical QT studies to the CSRC for research purposes. Next slide, please. So the FDA ECG warehouse, again, this was the one that was for regulatory use. It contained de-identified annotated ECG waveforms obtained in clinical QT studies and was stored in an external warehouse and used during regulatory review. So what FDA reviewers would do, they would access this warehouse online to verify the QT measurements. We would look for whether there was any signal quality, as well as any bias in the measurement. And then also the each of the digital ECGs was could be linked to the subject level variables such as demographics, treatment group, or drug concentrations, to be able to to link that the waveform to these other variables. And again, as I said, the warehouse was for many years located external to the FDA, but was transferred to the FDA in 2021. Next slide, please. Now, the other database, again, is the CSRC ECG warehouse, database, and this was the voluntary database. Now, the CSRC is a public-private partnership to advance knowledge on cardiac safety issues and sharing of ECG data for biomarker development is central to its mission. And so the way this would work was the CSRC formed this ECG database, and as sponsors of clinical studies agreed to share their data, they would authorize the FDA to release their ECG waveforms from the ECG warehouse to the CSRC database. And then the CSRC would then maintain the database and provide oversight for data release for research. And so if you're an investigator who would like to access that data, researchers would then submit a proposal, which would then have to be approved by the oversight committee to evaluate the stored ECGs. And so, so the CSRC did maintain a tight control over the use of the ECGs for research purposes. Next slide, please. So the FDA strongly supported this data sharing efforts for cardiac safety. For over 10 years, every time that we would review a QT study protocol, we would send companies the same comment, which is shown on the slide, about asking them to share their data with this, send their data to the CSRC. And even if they were hesitant on sharing their drug data, they could always share the placebo and positive control data from their QT studies. And if a sponsor agreed to share their data, they could release, the FDA would release both the waveforms from the warehouse as well as the study variables to the CSRC ECG database. And we also had a data request letter that a company could fill out, which would trigger this sharing, a releasing of their data to the CSRC. So next slide, please. So I'm going to go a little bit about the outcome of these two data sharing initiatives. So for the FDA ECG warehouse, again, this was for mainly for regulatory review. It ended up it has more than 13 million annotated ECG waveforms from over 1,000 QT studies. And these ECG waveforms are for placebo, baseline, in a diverse set of investigational treatments. And it also includes waveforms from both healthy volunteers and patients. And if we look at the CSRC ECG database, which is the voluntary data sharing database, it had only 20 QT studies with a little bit more than 100,000 annotated ECGs. We really couldn't get any company to submit their drug data. They did, from those 20 QT studies, what companies did end up submitting were waveforms for the baseline, the placebo, and positive controls. All 20 studies were in healthy volunteers, so we didn't have any patient data in the database. So you could see without a really strong support within the community that we really couldn't get a lot of companies to submit the drug data. Next slide, please. So what did we do with these databases in terms of advancing cardiac safety? So the FDA ECG database, which what it was used for was developing tools to read ECG biomarkers from the waveforms. It also was used to look at new biomarkers to evaluate mechanisms of action of drugs, as well as the potential for proteolytic risk. And additional tools were made to assess the quality of the measurement in terms of QT bias for sensitivity analyses. And the research that went on within the FDA using the warehouse generated 8 scientific manuscripts. If we look at the CSRC, because we really just couldn't get a lot of companies to support data, I think the scientific value of that database was lessened, but it was used to evaluate commercial ECG algorithms to improve standards for ECG measurements. And this database then generated 4 scientific manuscripts. Next slide, please. So I just want to kind of summarize some of the, what I would call lessons learned. And they're not just my lessons learned. I actually talked to folks who've been in this space for quite a bit of time, like Dr. Norman Stockbridge, as well as some of my colleagues on the IRT, in terms of what they thought was really important with these 2 databases. So I think the most important one is standardization is critical for any data sharing opportunities. In this case, we had an external group, HL7, develop a standardized format for the annotated ECG waveforms. And importantly, the stakeholders agreed to use this standardized format. Now, despite the strong support from the FDA and very enthusiastic stakeholders for data sharing initiatives, as well as our attempts to address some of the barriers to data sharing, there was really limited success in establishing the data sharing repository outside of the FDA. As I said, few sponsors released their ECG waveforms to the CSRC from their drugs, which really limited its scientific scope. And there could have been some issues with maybe how CSRC stewardship of these data may have constrained some of its use. Now, submitting data to the FDA warehouse as opposed to the CSRC database, I think that was a little bit easier because companies were already submitting their data to the FDA for regulatory review. So they really eliminated any types of concerns about confidentiality or legal or ethical concerns about how the data would be used. Next slide, please. So I think that the ECG space is unique and might be different than for the drug-induced kidney injury space, because FDA was able to leverage a safety guideline, which is mainly the E14 guideline, to request sponsors to submit the annotated ECG waveforms for both approved and investigational drugs and biologics. And any initial concerns that the FDA might find a new safety signal in the waveform data really didn't come to be. The regulatory review was confined generally to the guideline E14, and the annotated waveforms are really used to look at quality of the QT measurement. We weren't looking at any other kind of biomarkers within the waveform during regulatory review. Whereas the scientific focus of the ECG warehouse on new biomarker development or tool development for cardiac safety was done outside the regulatory review. Also, what we did at the FDA was we used the established regulatory science program to hire full-time fellows for research purposes. And they're the ones that really advance the science in this space in terms of developing new cardiac safety tools, and as well as evaluating the ECGs for new biomarkers from the drugs in the database. And also all the tools that were generated based on the databases are publicly available. Now, we did look for and propose that external companies could develop widgets or some type of approach to submit code to get data out of the warehouse without actually seeing the proprietary ECGs or maybe just getting aggregate data out of the warehouse. But we really couldn't get companies to really have much interest in this type of approach to data sharing. So this really didn't, I don't think we have any examples of where a company was able to submit codes to analyze the warehouse. So that was the lessons learned from the ECG experience. And I'd like to acknowledge my colleagues, Dr. Norman Stockbridge, Lisa Thompson, Jose Vicente, who is now managing the ECG warehouse at the FDA, and Lars Johansson, as well as some colleagues at the CSRC database, Cindy Green and Valerie Morrow. So I'd like to thank them for their contributions to this slide set. So thank you. And I guess I'll be talking with everybody during the panel discussion.
Thank you very much, Christine. All right, well, I'd like to introduce our final speaker today, and this is Dr. Shashi Ramaya. Shashi is a PhD veterinary scientist and a board certified toxicologist and pathologist. He is currently a vice president at Pfizer and the head of multiple discovery, investigative, and translational lab groups at Pfizer. Additionally, he is the industry leader for the TransBioline Consortium, and that is working on advancing safety biomarkers and clinical qualification of those biomarkers. So Shashi, just let Katrina know when to advance the slides, and pass it over to you right now.
Okay, thanks, Nick. So I'll try to see if I can wrap this up quickly. I know we are probably running a little behind. So first off, I want to thank the organizers, the PSTC, for this invitation. And really, I also want to congratulate and thank all of you to, especially the organizers for bringing all the outstanding stakeholders here, right? The regulators, the industry, and most importantly, the patient voice. It was loud and clear, bringing to the front on all the needs, etc. And also thanks, Amanda, for highlighting the value proposition of data sharing and the motivation behind it. Definitely makes my job a little easier as I advance these slides. So what I want to do today is maybe touch on a couple of things. One is about TransBioline, this large consortia where there's a really good opportunity to share broadly some of the data sets, specifically with CPAT, the data repository we've been talking about throughout the day. And I will highlight to you some aspects of clinical cohorts, kind of data sets, regulatory interactions, and focus on, of course, the kidney work package. In the second half, what I want to touch on is some areas we are proactively working within Pfizer to be able to transfer data sets as we generate internally. We have some case examples and some success stories, and hopefully we can use this opportunity to be able to work on future concepts and cases as well. So first, if you go to the next slide, this is really a broad stroke on the introduction to TransBioline. This consortia is an IMI consortia, which has got multiple academic partners, 7 industry companies, that's focused on 5 organ packages. That's the focus. And at the end of the day, it's about generating data sets for regulatory qualification. Of course, in terms of deliverables, we want to be able to have the assays for all these work packages. And these are consistent to the framework that FDA has established. And obviously, to be able to work on these organ systems and the assays, we need the right type of cohorts, clinical cohorts that we are planning to derive from. And finally, our intent is to be able to apply these biomarkers and concepts to our ongoing clinical trials to mitigate safety issues. So that's the sort of the high level on what TransBioline is. And the next slide talks about the assays. We have close to 50 biomarkers across all these packages. And as you can see, these are all pretty much, I would say, 90 to 95% been validated and ready to go to test these biomarkers in our samples, clinical samples. And I want to point out that for kidney, we have glomerular injury biomarkers that are listed here, as well as tubular markers that we've been talking about both from an individual as well as the composite biomarker standpoint. So a lot of work, but these assays have already been validated or near completion from TransBioLine. If you go to the next slide, this slide in one view depicts where we are in terms of regulatory qualification. You know, that's the 3 steps we talk about, right? The letter of intent, the pre-qualification, and the qualification plan. Qualification plan and full qualification package. So as you can see, for all these packages, we have completed the LOI submission and is accepted into the biomarker qualification program within the FDA, except pancreas, which is ongoing. From an EMA standpoint, all these work packages have been accepted and the qualification advice has been received. So specifically, work package 1 is drug-induced kidney injury. That's the package where we have both FDA and EMA acceptance. If you go to the next slide, this slide, again, in one slide, talks about this large effort around collecting patients' samples as well as concurrent healthy samples across all these 5 packages, right? Specifically, I want to highlight the first one. Work package 1 is the kidney package. That's around glomerular injury. So for this, as you can see, we have a plan to test these hypothesized biomarkers in the learning phase cohort, and then healthy controls. As you can see, we have concurrent controls, and then based on the preliminary data, we want to test them in the prospective clinical trial samples that are ongoing. And as you can see, that's been a little challenging and a little slow, but overall, making great progress in recruiting, collecting, and testing these samples with the biomarkers I mentioned. And imagine a scenario where generating these data sets for kidney biomarkers in these cohorts. It will be incredible to have these set of samples broadly shared across the community and specifically within Cpath. So the next slide talks about glomerular injury. So I'm in the interest of time, I will not elaborate on this. All this I want to tell you is that the context of use for kidney package is around developing glomerular biomarkers as individually or composite to be used for the detection of glomerular injury in early clinical trial subjects with normal kidney function. So in the next slide, again, to show you, we have both podocyte biomarkers as well as tubular injury biomarkers that will be assessed from all these samples, and the technology is being implemented and the assays are close to completion as well. The next slide talks about really at a high level, 2 clinical protocols that's been employed for a confirmatory phase. One is an anti-VEGF protocol. On the right is the preeclampsia. As you can imagine, we have really good set of sample sets that will be available. And the left side, we have patient population both with and without anti-VEGF, as well as categorized based on the UPCR as well. So close to 120 subjects in the VEGF, as well as the preeclampsia-induced hypertension protocol. So you will have these 120 subjects where you will have all these biomarkers, glomerular injury biomarkers, as well as the tubular injury biomarkers that will be available. If you go to the next slide, So how do we envision this? Again, this is in a concept stage, but there's a lot of groundwork being done already with TransFireLine to share. I think this may be a build slide. I didn't mean to, but anyway, the data ultimately will be available. The lab data, the clinical data, all will be available in Transmart and our own cloud for us to be able to generate the data sets for qualification. But again, the opportunity here is to share this data broadly with the biomarker data repository that we've been talking about. I'm not going to, expand on the value proposition. Amanda did a fantastic job on the value of broadly sharing these data sets. If you go to the next slide. Let me pivot a little bit, and that's the TransFireLine. Now I want to quickly touch on Pfizer, what Pfizer is doing to share broadly. One example, I have actually two examples. One example is the recent sharing of the Duchenne muscular dystrophy clinical data sets from an internal Duchenne muscular dystrophy program. On the left, you see some of the data sets, the efficacy biomarkers, the lab data were already shared with the CPath as part of the de-risk Duchenne regulatory consortia within CPath. There's already a data contribution agreement already completed. And so we want to be able to leverage this agreement that is in place already so that we can share additional sets of biomarker data that we may be generating specifically with respect to kidney, as I will share in the next slide. And just staying on this slide, you will see again, there are some aspects that we have not shared yet because obviously some challenges with different databases and the timing of arrival of these data sets is challenging. So because of that, we've only shared data from the clinical database, but additional follow-up around muscle injury panel is ongoing, at least for this Domacrozumab Duchenne muscular dystrophy project. So going to the next slide, this specifically talks about some of the ongoing work in terms of implementing these biomarkers prospectively for our internal phase 1 and potentially phase 2 and later on clinical trial, where these assays that we've talked about has been already validated internally. We were able to implement these biomarkers for a couple of projects already, Project X and Project Y, and we have data available for all these biomarkers. As you can see in Project X, across different groups, the different biomarkers, how they are performing, and as well as in towards the end, Project Y, as you can see in the composite measure, you can see a clear signal in group 4. So again, just to share an example of how we want to leverage the previously shared DMD data set and see how we can use the same process for us to broadly share this data set. Just to stay on the slide, we also have plans to understand these biomarkers in healthy volunteers across different disease areas. So all these are in progress, and we are hoping that such data sets will be valuable to broadly share and recognizing that there will be some real value in generating insights into these biomarkers. So if you go to the next slide, I think hopefully I shared with you the value and the large data sets that's coming out of TransFireLine, in addition to help mitigate clinical safety concerns in our own clinical trial. Hopefully we can broadly share this data set. I shared with you some biomarker data sets in DMD and renal biomarker areas where we can begin to populate the biomarker data repository and obviously support the commitment to patients. And importantly, I think all this will have some challenges. So obviously, leadership from the FDA, CDER, CDRH, and of course CDER and ODS Jeff Siegel and others. We've already been engaged in the Transboline Consortia, CPath, of course, and the leadership in Transboline is going to be extremely critical as we help navigate this space of broadly sharing these data sets. So before I close, I also want to acknowledge, several colleagues internally in Pfizer. I'm sure I've left out a few of them, but these are the people, that are pretty much engaged in championing and generating these data sets internally, as well as thinking about how to broadly share the data. So with this, Nick, I'm done with my presentation.
Great, thank you very much, Shashi. That's a very exciting set of data that you're looking to contribute right now. We hope the first of many contributions, and look forward to working with you on this. Well, at this point, we have one more video to show today. And so, Katrina, if you'd like to bring up the video.
I think data sharing is important, not only to ensure that we have drug safety and efficacy, but also so that we can develop or identify new biomarkers, not just creatinine, that will help us anticipate drug injury earlier. Currently, creatinine is used for just about everything with kidney disease. But if we could identify some other biomarkers that were perhaps more nuanced, that would signal the injury earlier or the potential for injury earlier, we could perhaps prevent the occurrence of drug-induced kidney injury.
Data sharing is important to me because it gives me hope. It gives me hope that if we could put enough good, solid data in front of these companies who manufacture the drugs or who come up with the research and development, there could be hundreds of thousands of people who will live or not have to live in fear of dying because of their kidneys.
In the course of any and every clinical trial, there is the all-important standardized and novel biomarker data collection that goes on. When you group different organizations together and bring their specific trial data together, there may be points where the information provides greater support or non-support for a conclusion drawn in one study as opposed to another study. Altogether, the sum of the whole is greater than the sum of the parts when you take one organization studies versus another. Those individual studies carry just so much weight. When you pull all their data together, the information may be much more compelling.
It'll be important for individual patients who might be experiencing kidney injury to allow the use of their data so that we can build on their experiences and share the knowledge across the wider group in order to help these efforts. Of course, those data will only be shared under certain very strict conditions, i.e. the patient should consent to allow us the use of those data, and also that we would never be able to identify individual patients from their data.
I would like my identify you share because I want to help. I want to be a part of that, be part of the pioneers that are working hard to make this happen.
I want my data to be shared, and I hope that many, many, many patients share their data as well. And to me, I feel safe because I know that my name and my other information, personal information, is not associated with that.
It will be important for numerous pharmaceutical companies to come together and share data across the consortium in order that we can pool together these data sets and create a larger database that we can then interrogate in order to identify and to validate these novel safety biomarkers. Individual companies will never be able to go it alone with regards to collecting enough data.
By contributing data, we're only going to be able to improve our methods and modes of treatment. By not contributing data, then we're withholding valuable information that could be used to help impact the future of patient care.
Data sharing is critical. There is no single study or source of information that tells us whether a biomarker, something we measure in the blood or maybe the urine, will perform well in detecting kidney injury. To understand whether a biomarker is a good indicator of kidney injury, we will need to integrate data from a lot of different sources and studies. That's why data sharing is so critical. We won't make progress unless we share data.
Thank you, everyone, for taking the time to make these videos and sharing your thoughts with us today. I'd like to move into our final panel discussion. As a reminder, again, those in the audience, you may ask questions via the chat tool. And again, this workshop is being recorded and will be made available soon after today. I'd like to invite the speakers from the current section. So if Amanda, Christine, and Shashi will join us. In addition to these three speakers, we have a few additional panelists I'd like to ask to join us right now, which includes Glenda Roberts, Robert Starr, Mike Pekanowski, and Ray Harris. All right, thank you everyone for joining us. And so we heard some compelling talks from people with various perspectives on successes in data sharing and also looking a little bit towards the future. Right now, I'd like to give those who haven't spoken just a moment if you'd like to share some of your thoughts, Mike, Ray, Rob, and Glenda, before we move into some of the audience questions.
So I'm not shy. I'll speak up.
Okay.
So I want to start as a person who is totally needle phobic to say what Amanda said. You know, to the extent that patients have an opportunity to share their data without having to go through multiple traumatic experiences with blood draws and that kind of thing, I think that it'll be easier to get patients to engage. And the other thing that I think is really important to help with recruitment is that industry and researchers look at developing informed consents that are patient-friendly. And by that, I mean, you know, when you give people 4 or 5 pages to read with a lot of legal jargon, it makes people less likely to participate. Because my experience with patients is that many patients are altruistic. We want to participate, we want to be asked, but we're also put off by having to sign what sound like complex legal documents that won't allow us to make the strongest contributions. So I have other thoughts, but I'll stop there.
Yeah, could I just sort of second what Glenda just said? You know, here at Vanderbilt, we have a big biorepository called BioView, and it was initially an opt-out, but it's now an opt-in. And basically, they just use sort of discarded blood to get DNA for patients. But I think they've developed very simple sort of consent forms so that it tells patients that their data will never be identified and that it will be used for hopefully to help other patients. And most patients will agree to that sort of analysis as long as it's not, and you know, it doesn't have to be difficult. All you have to tell patients is that, as I think it was one of the transplant patients said, she knew that her data would not be linked back to her. And so I think that's really all that is necessary to do this. I'd like to also ask Amanda, because we had in our breakout session, we had quite a bit of discussion about how to, the best ways to engage the patient to be the best advocate. And do you have like a very simple mantra that you would have patients so that they can really be the advocate to the doctors or to whoever is running the trial, what to tell them?
I mean, thank you for the opportunity to answer that question. I don't know if I have a simple mantra, but I will tell you that my team of physicians and nurses and healthcare providers, they're used to me pushing them to share. I mean, I obviously, when I saw the publication, I went and downloaded the data myself, and I don't know which of those records I am. So there's a plug for de-identification for patient information. I'm pretty much an expert, right? So I downloaded all of the data from Nature Medicine, and I couldn't re-identify myself. I tried, and I did try, by the way. So I think that's a good plug. But you know, I think it's twofold, Ray. It's not just patients advocating that study designers and owners share their data. It is partly that, right? But I think it also goes back to Glenda's comment about the legal jargon in the document, right? And I think I mean, from my own personal experience, Glenda, I think that part of the reason that that's off-putting is it looks very much like that legal document you're signing is built with the concerns of the study owner, not the concerns of the patient, right? So it feels very much like you're signing something that says, I don't have rights and you have you have ultimate rights and you can do whatever you want. And so I mean, I think just kind of summarizing, I would say that we should be building consents that help the patient feel empowered, that acknowledge that the patients own the data, that help patients feel more comfortable that their data will be kept private, but also in the other direction, ensures, gives the patient some reassurance that the data will be used to its maximum value and not put into a drawer and put away right after the study is done. Yeah.
Thank you all for those pieces. And I do also pause for one moment and introduce one additional participant on our panel right now. Dr. Norman Stockbridge from the FDA is going to be joining us too. Norm, is there anything you'd like to add right now around data sharing or empowering the patient to feel comfortable in sharing data?
Yeah, I just wanted to note that, you know, we've heard a lot of success stories the last couple of days. There is a fair amount of sharing going on. Patients are saying yes, companies are saying yes. You know, there's a fair amount of data at CPath, you know, compiled and available for use. I'm wondering where people actually think the rate limiting step is.
So, hey, this is Rob Starr. If I can say a couple things also. I want to start in one direction, but end up what Norm just said. I think that's really true. As we put together the Kidney Precision Medicine project that you've now heard about a couple of times, we realized that There were scientific issues, there were clinical issues, there were safety issues, but the real problem was a cultural issue of nephrologists not doing kidney biopsies. That 40 years ago, nephrologists had decided we don't need a kidney biopsy, we're not going to learn enough, we already know what we need to know. And that's what KPMP was really pushing against. The mantra that we've used, and Paul Kimmel, who's on here, is the one who I think developed it with Richard Knight, is patients or participants first, safety first. And that's how we put this study together. The cultural problem of the kidney biopsy issue was attacked, A, by showing that it was ethically permissible to do it. The participants were lining up. We had a meeting, I remember KHI meeting, I think it was, where patients were lining up saying, please, please do this because of the tremendous altruism that you've heard about. So to me, Norman's question is important because from drug companies, yes, we need to understand why you're on the fence. And what is it about the value proposition that's not making sense to you? But then thread into that this concept of goodwill and being part of the commons. The other problem here is this is so-called tragedy of the commons. You're trying to develop a common project where everybody will benefit, but nobody wants to pay for it. I know this, that the NIH is paying about $120 million for KPMP over the course of the first 10 years of the project. And we decided that we're just going to do it. It needs to be done. We need to do it. We'll pay for it. And we hope that people will join us. But the third point, which I understand from the industry side, is timelines. These timelines are so much longer than the 3 years window of opportunity that you're used to all thinking about. We understand that, but that's the reality. And that's, you've seen now a couple of examples of the timelines which span more than 10 years to get something done. So that's what it will take. And I do not want to overpromise anything.
Mike, I see that you have your hand raised right now.
Yeah, so I sort of build on that a little bit. My name is Mike from Office of Clinical Pharmacology at FDA. I think we've heard a lot about sort of the data sharing and whatnot through the lens of trying to pursue qualification and aggregating data across many data sets and many studies. And I think there's an element of data sharing that perhaps is a little bit different and much more micro in its nature. And that is sort of the day-to-day work that happens between companies and FDA with respect to specific drug development programs. We work with companies routinely on endpoints and spend a lot of time determining what kind of properties it needs to have to support surrogacy and Subpart H approval and so on. And we learn across different programs as part of that process. And I think one issue or gap, at least perhaps in this space, is we may have a data generation issue where there's still a reluctance among companies to generate these data because obviously it would potentially threaten the program. But it also means that we are not learning in that little microcosm as well about the performance of these biomarkers as they relate to the preclinical signals or eventual damage to the kidney. So those, it's something important to think about a little bit and whether there are workarounds or ways to overcome that issue, because companies aren't going to be incentivized to generate and submit data to us. We don't have the benefit here, as Christine was discussing before, of guidelines to produce these data as part of drug development.
Thanks, Mike. And Shashi, I'm going to kind of come back to you. I realized that we didn't quite balance across all stakeholders here, but I'm going to call on you right now, Shashi, just in terms of, for what you just proposed in sharing, what was the tipping point within Pfizer in discussions within your team and with your senior leadership of beginning to share data like this? What was important and compelling?
Look, I think the point about overpromising kind of hit me a little bit, right? It's generally people are willing to share, right? They're, I think if you talk to people, if you showcase them the right science, right questions we're asking, people are naturally willing. The challenge is always the process, right? The process, people always lean on things that are easier for them, right? So that's one piece that we can definitely work on, more educating people, right? And second thing is about championing someone and being a champion internally and showcasing that, look, the data is good and we have less of a challenge to be sharing the data set. It'll help the broader community. People get it, right? So I think, and also sometimes, when drug development programs, sometimes when, it gets terminated or whatever, for some reasons, it makes it more easier. But I think there's still some nervousness about all the stuff that think about why it's difficult to share, right? Amanda, I think, maybe I'm going to go back and listen to this again, because it is so real. Those questions she highlighted are all real. And I think, it's good to see that it's not as bad, right? So I don't think I gave you a sort of specific response, but I'm just laying out some general concepts as to how, people think about.
Christine, with CSRC and the number of studies that transferred from within FDA into the CSRC database or warehouse, do you think that that's something that was a little bit of the time, and that if you were to begin to pursue at this point where you are seeing more data sharing across different areas, that it may be something that may open up more so in the future?
Yeah, I think that I was actually really impressed with Amanda's presentation. I mean, we didn't have a champion like Amanda going and talking at cardiac safety meetings about the importance and how to overcome some of the barriers of sharing data. So I think if we actually had Amanda come to our meetings and talk about doing it and what we're going to do with the data and why it's important to share, I think we probably would have gotten more companies to share. I don't know, Norman, what do you think? You were there in the beginning, and it was really your brainchild to think about developing this warehouse. How do you?
Yeah, I was pretty depressed by the experience we had, which you well know. We got about 1% of the data shared, our efforts weren't targeted at, these are mostly healthy volunteers. They weren't patients in these studies. They're mostly healthies. And so our efforts were all targeted towards the companies that were providing us the data. And for years, every communication we had with them, every written communication with them, had a paragraph in there saying how important we thought it was for people to share their data. And it basically did not happen. In important ways, it just didn't happen.
I was also really impressed with having the patients coming on in this group and talking about the importance from a patient's perspective it is to share data. So I think what I'm seeing is, I like what you're seeing, and I hope your initiative is data sharing is very successful. I think you're doing all the right things.
I'd actually like to build on the comment that Christine just made about the importance of bringing patients to the table. Like, that's the buzzword right now. Everybody's talking about it. But I think as a consortium, we have an opportunity to model the way. I think you have an opportunity to bring in a patient engagement group that is actively involved with your consortium, that it is important to have a patient that's a part of your leadership team, that committee that started off being the steering committee. I think that demonstrating how you can effectively engage patients within the consortium would represent a good model for industry, because what I'm learning from academic research, and Paul Kimmel and Rob Starr can both attest to this because I work on 2 projects that they're both involved with. When we bring patients to the table and engage them with researchers, the outcome is that even if the patients are quiet, eventually the researchers will turn to the patients and say, "What do you think?" And it is also important that the patients look like the people that your drugs are intended to serve. And I can't encourage you enough, as you start to develop your own patient advisory boards, to promote and advertise the fact that you have patients serving in this capacity. Put those pictures out there so that people can see that there are people who look like them that are actively involved in your research. And I think that it will help with your recruitment efforts.
I have a question, I guess, to the group. Some of this conversation is reminiscent of, I think, what happened in cardiology, where I think it was the academic researchers, but I'm not sure who, maybe it was the FDA, decided that they were no longer going to accept placebo-controlled studies in angina, and that you had to have active drug comparator studies. Somewhere in the community, people got together and said, we're not doing it the old way anymore. Does anybody have information on that, either Norm or others? Christine?
Pause for a moment to see if someone in the audience has a recall on this too.
Or other situations where there really was a cultural change that took place that led to an outcome that was far better for everyone.
Amanda, perhaps with the Alzheimer's work, I know that initially that was all placebo-arm data that was being contributed, but over the past few years, we've expanded that database that's been collected. Has there been a shift in the data being contributed? Is it still solely placebo-arm, or have they begun to share treatment arms?
I'll be careful because I haven't been as actively involved recently in our Alzheimer's consortium, but my perception peripherally from talking to data managers working on those data is that yes, there has been a shift. And so, I talked in my discussion about some of the ways that we arrange data use agreements and data collaboration agreements, Nick, and one of the ways that when we reach a customer, I'll call them a customer, right? A data owner at a pharmaceutical company who has a really honestly valid concern about sharing competitive intel that might help their competitors have an advantage, right? What we always fall back to at Cpath is this idea that you can share data with Cpath. We're a neutral convener. There's no risk. We're not going to compete with you. And we are able sometimes to get access to really highly valuable studies or data sets or trials that they would not be willing to share with their competitors, but they're willing to share it with Cpath. And so my impression from the latest activity in Cpath is that there are 2 or 3 really high value studies and trials that have come in that are not shareable external to Cpath, but that everyone who shares the data will have access to open source tools like markers of disease progression, for example, clinical trial simulators that help you enrich for particular subsets of patients. So everyone has equal access to the tools that are developed as a result of the sharing, but it helps to de-risk this feeling that sharing the data might actually cost my company or my research. One of the things I will just add to close that thought is that many times I have seen people who are completely unwilling to share, and the only way to move that needle is to get them to share with just CPath for the purpose of producing a model. And then they eventually learn through what I call the democratization process, peer pressure. So what they do is they eventually learn that there's nothing that's going to get, you know, if a company doesn't have a molecule in the pipeline already, they're not going to suddenly go, oh gosh, I saw that one study and now I know how to get ahead of, you know what I mean? So what we have found is that we will encourage people to share as openly as possible if they're only willing to share it internally with Cpath in order to develop a biomarker model. Then we go back to them and we say, okay, now that you've shared it and you see this progress, and you know, would you be comfortable increasing the level of sharing? And so I think honestly, the carrot approach kind of helps. And then having, when Shashi said, hey, Pfizer's going to be sharing all this incredible data, that's another example, right? A little bit of peer pressure in a positive direction that says, when you, Nick, are going to another company, you can say, well, you know, Pfizer thinks it's okay. Pfizer is sharing this incredible data set. It puts a little bit of extra pressure, I think.
And we have a question for Mike right now. So the FDA had a voluntary data submission program for genomic and exploratory biomarkers. Can you talk a bit about that program and any learnings that can be applied in the drug-induced kidney injury space?
Yeah, sure. I think you know, that program is something that was launched back in 2004, 2005, and you know, was initially focused on genomics data, but sort of later expanded to other types of exploratory data, whether non-clinical or clinical. And it was really a forum for non-regulatory interactions between pharma as well as even academic partners to discuss data. So genomics was, you know, relatively new at the time, and there was a fair amount of uncertainty about how FDA would be reacting to those data and the results generated from those kinds of studies. So, you know, this really created a safe harbor, for lack of a better phrase, to talk about the data and even address some logistical issues like how to submit the data, because, you know, there are large volume data sets and things like that raised some new computational challenges for FDA. So, you know, that I think, you know, was a good forum, you know, for looking at subject level data. We would do analyses and talk about the results. It later became such that, you know, we began talking about trial results, and it was much less in that kind of one separated space from the drug development program. So, you know, we saw a transition into, you know, those data coming in through the regular regulatory channels. And that I think that forum for non-regulatory engagements largely been replaced by critical path innovation meetings. But nonetheless, I mean, I think, you know, as it stands today, you know, there was benefits to that program. It provided some protection from the companies about worrying about how we would react to their data, or we would put their program on hold. You know, so it was useful. You know, and I think apart from that, you know, now there probably still exists large, massive amounts of data that never sort of make it to FDA. They're not required to be submitted, you know, per the regulations. So there's a lot of data that may not see the light of day, and a lot of those concerns about us overreacting, you know, might still remain as well. You know, but I keep thinking in this context, at least, it could be, you know, when these kinds of data are discussed with us, that, you know, it might end up telling us that the biomarkers don't perform well, and in the future we won't overreact to them, or conversely that they perform so well that they could rule out signals of toxicity and things like that. And I think that those positive outcomes need to be kept in mind. Sorry, I digressed a little bit.
Yeah, we've heard lots of companies cite concerns about us overreacting to data. You know, there had been some notion early on about having a quote safe harbor where you were guaranteed we weren't going to react adversely to what we found. But there was no way to ensure that. You were looking for safety markers. If you found a safety signal, you had to do something with it. So it is part of, I think, why companies are inhibited with having us share data. Not sure what the solution to that is.
If I could just add on briefly as well, I think in that vein, BioView was mentioned earlier, as a model for data collection and data generation. And I think there's probably great promise for academically driven studies and, healthcare institutions. We now have tremendously large biobanks that are linked to electronic health records. And, I think it presents a great opportunity to start skimming biospecimens, if you will, from ERs and other areas where you can link those data to outcomes and start getting some better information on the validity of a biomarker to that point.
A question for Norm, because I understand, completely what you said about why companies would be worried, but if there is a safety signal, wouldn't a company want to know it as early as possible? Or they just want to get the drug on the market and then see what happens?
Well, if you believe in your drug, you're apt to worry that some signal will show up that gets more attention than it deserves. The agency caused people to spend I don't know, something around $1 billion on QT studies, and diverted drug development away from drugs that had a potential QT liability. In doing so, they got, they weren't optimizing for the intended effects of drugs, and they were probably inheriting other toxicities along the way. So it's not entirely irrational for them to worry about the kind of response that they get when a safety signal gets identified. I'm not sure we, over the last 20 years, did the community a total favor in cutting down on the amount of proarrhythmia risk there is in new drugs. That is not an official FDA position.
We appreciate your perspective, Norm. No, and I was curious if the term safe harbor would come up during this discussion, because it seems to be one that always comes up. And I think the way you put it is if there's a signal, it has to be addressed in some way. But in some manner, it may be how that signal is addressed as something better than there's no reaction to a signal, but it's beginning to discuss what types of steps and flags are put in place with exploratory biomarkers when you see changes, rather than saying safe harbor, but putting in place different appropriate steps to address and understand these changes when you see them, rather than thinking that it's going to be a full stop reaction.
Nick, if I can chime in too, just again, as a patient, I'm thinking about a particular biomarker that we use in my progression, and it's so frustrating because it's meaningless for me as a patient. It just curiously jumps up and down with no, and so it has caused me to be overtreated, honestly, because a blood biomarker will spike up and then I'm like suddenly not able to stay on my regimen, or, all these issues that patients have. So, I mean, I think the other the point that I just wanted to make based on what you were saying is, if there's some sort of sense that reporting safety issues is okay because it might, the fear is reporting a safety issue might cause this knee-jerk reaction that burdens companies to, prove safety. The other thing that it might do, though, is help you to optimize and refine a biomarker in such a way that it can address the issue that I have, which is better, more optimized safety biomarkers might also help you to continue a trial safely, because that, like, for example, creatinine, Glenda brought that up, it might not be indicating the kidney injury. It might be a Chicken Little response, right? Because we have these other better biomarkers that help us to know that we can keep the trial running and we can keep patients safe. So that's a part of the value that has to be considered here, is that sharing data to optimize better tools, better biomarkers might also help you to know that when, of course it's affecting kidneys, but it's within a safe window that interventions can help still, right? So, I do think there's the potential for data sharing in this particular area to help safety biomarkers become optimized in such a way that things that in the past might have been halted based on safety concerns might actually, in the future, with better biomarkers based on data sharing, might actually help you to continue and optimize, or enrich for patients that identify patients that might be particularly vulnerable that shouldn't be taking that drug, without eliminating that other larger population, right? Yeah, so I think that's a part that's important to talk about as well.
Yeah, definitely. It's kind of not the predictive component of understanding, and predictive in the sense of understanding people at risk, at a higher risk of developing that type of injury.
Yeah.
And also we've talked about, these exploratory biomarkers as we continue to learn more about the biology, the performance, right? It's not that you see signals, it's going to be like, oh, there's some real safety issue, right? There is also additional elements that you got to look at. You're not going to terminate the program. I think someone, I don't know who was talking about, maybe a patient was talking about yesterday that, just because one goes up doesn't mean that it's an issue, right? You look at the overall patient, right? There are other endpoints that are collective evidence you gather to show maybe there's some educational piece too, the community as well as some drug developers, to help understand that, look, we are still in a learning stage. So let's not make a decision just based on some of these exploratory biomarkers.
Yeah, thank you, Josh.
Just one other comment, maybe to follow the line of logic around overreacting and whatnot. I'm just thinking out loud, wonder if there are opportunities for specimen sharing too that could potentially be thought of. That would, there could be data safe harbors, but then data could also not even be generated and put to some third party where they could be assayed and processed and in a way that's potentially not offensive to anybody, but still gets data linked to the clinical information.
Yeah, no, I know that in the breakouts, two suggestions that I heard that I think have potential in terms of looking prospectively is one, the American College of Medical Toxicologists has a program, a consortium within there that collects samples from people who have some type of toxic reaction to a drug or something around the household, and they're embedded throughout the country in a lot of underserved communities as well. And so that's one potential opportunity and way of obtaining samples in the future. The other is that in emergency rooms, the observation that a lot of times the majority of at least like the blood samples or urine samples that are collected, only a fraction of what's collected is analyzed and the rest is discarded. And so those discards can become very valuable samples in the future.
Yeah, that's exactly what Vanderbilt does with it, just discarded blood to build their BioView, which has like 700,000 patients or something like that. So it's a very easy thing. So any patient who comes in the hospital for any reason or goes to the clinic for any reason, you know, as you say, they draw a lot more blood than they need for the sample.
Well, do you mind if I expand on that with you, Ray? So what did it take in terms of implementing that? You know, just a few components of what led to a shift and requesting that of people coming in.
Well, initially it was a opt-out for probably the first 10 years of it, or 8 or 10 years, and then I think the some ethics committee decided it should be an opt-in. But I think as we're talking and what sort of a follow-up to what Glenda had said, it's a very simple form that does not require a lot of you know, in-depth analysis of whether, you know, you're going to be known or anything. All it says is you're going to be anonymized, and this information will be used for scientific purposes to help the benefit of other patients. But you will probably not get any direct benefit per se. So it's a very altruistic thing.
It's kind of similar to going to the Red Cross and donating your blood for future use, but in this case, scientific.
Exactly, and you know, the Red Cross continues to get blood samples for blood donation for people that know it's not going to go to them necessarily, but it may help somebody that they know. So, and that's the same way that altruism works, hopefully.
All right, well, I will see. I think, oh, we have one additional question that just came up actually for you, Dr. Harris. Are the biosamples at Vanderbilt linked to patient clinical data? And do you have examples of how these samples are being used?
Yeah, they are definitely, because they are linked to an anonymized database. It's called the synthetic derivative, where all patient identifications are changed so that there's no really minimal, if any, chance of ever going back to find that patient. But what it allows is the full medical record, as well as the medical record going forward after the sample. And it's been used quite extensively by researchers at Vanderbilt, at other institutions, and actually now pharma is very involved in using it. One of the specific aspects that they're using is something called a PWAS, where they will take a SNP of a specific gene, which may be involved in a disease, a known disease, and look to see whether it may correlate with other patient outcomes, so that they've identified a number of different conditions where a gene which is known to affect one disease may actually affect some other disease. And so as a potential for drug development, it's really very, very helpful.
That's interesting. And I think there's also a request of you, Ray, if it's possible to share some of that, the language in your consent form that you use.
I think what you probably should do is contact the Vanderbilt BioView directly, because I'm not directly connected to BioView, and so I don't know exactly whether they would agree. I'm sure they'll agree, but I don't have that.
Thanks. Okay. Well, we have a few more minutes. Are there any additional questions from the panel for others on the panel? Oh, you're on mute right now, Josh. There you go.
Yeah. So, you know, I'm just curious about the concern about, you know, the generating data that might be sensitive, that might impact the drug, et cetera, right? So how do we educate this? Is this, you know, I think all the stuff that people talked about, Amanda, it's all real. So while you have put all that in one slide, I'm not sure, if this is, this is purely an educational thing. So is there an avenue where you could do more of this, publications and opinion papers, and there's got to be some way to help digest this into drug developers as well.
Yeah, well,
No, we do that, don't we, Nick? I mean, we do a lot of advocating for data sharing and publishing of papers, Shashi, and we actually have an FAQ on data sharing at our Data Collaboration Center website, and we have case studies that we publish from time to time about data sharing. There's a list actually at the CPath website of data sharing papers and things like that, where we've talked about the benefit to public. I think that probably it's just continuing to toe the line. I think it's education for sure. The people that we run into that are still resistant, though, it seems to be more cultural than education. I mean, they've heard all of this stuff. Sometimes what happens is that it needs to feel contextually real for an individual, right? Because I'm so nervous about, I haven't done this before, and I feel really protective of my intellectual property, and I'm nervous that it's going to give my competitors an advantage, or others are going to find things that I didn't find. It's going to highlight that I missed something. These are kind of the common cultural concerns that we hear. Until you've actually gone through the process, you can't really know and deeply understand that it's safer than you think. It's probably just, all these fears have been addressed with other people. I think that what will move the needle, though, is greater mandates. So in academic study, grant funders are mandating open sharing. They've gotten smart. I mean, one of the things that I've seen is that NIH grants will mandate fair data sharing, and there were some people who were ticking the box without actually sharing anything useful. So they've gotten smart about being very explicit in what they mandate. We will fund you, but you will share the product of your research in the following ways specifically. I think that's an important piece of it. But from regulatory agencies, I think that's the incentive that pharmaceutical companies need to take that first little, put the toe in the water of sharing data a little bit. And it's probably two things. It's probably, guidance and mandates and things like that, but it's also probably this, I hate that phrase, Nick, I'm going to use it, safe harbor again, but it's probably some process for pharmaceutical companies to feel as though they aren't putting themselves at risk by sharing data. So some sorts of open dialogue or discussion or guidance that helps companies know that if I share under these conditions for the sake of advancing safety for everyone, that's not going to be, you're not going to be penalized for that. I think you're right about education, but you can only say it so many times to the same individual. It really, what else is there that needs to give them that little push? Do you want to add to that, Nick?
Well, the piece that just came to my mind in terms of education is where we educate somewhat. There was an article over the weekend in the New York Times about APOL1 gene and gene variants of that are linked to increased risk for kidney disease. Very apropos to see that this week before the meeting. But that's an avenue of education that I think that is kind of the next step of taking it to a broader population. It's something that people at all levels of companies are going to see a New York Times or Washington Post article, and when you start to see pieces like that popping into the culture as a whole, that's another way to move the needle forward.
So what you're saying, Nick, is just after the meeting, everyone will not start sharing right away. Got it.
Or they'll call the New York Times, either one.
Yeah. Yeah. Well, I think that we have talked through this component significantly over the past 30 to 40 minutes. And if there aren't any additional closing comments you'd like to make right now, we can move to closing today's workshop and the end of the conference. So I'll pause for a moment if there's anything someone on the panel would like to close with, and then I have a few final words.
Okay, I'll close.
Go ahead, Linda.
You know, we make light sometimes of the importance of data sharing to patients and how patients have fears and sometimes we're reticent. But I can't say enough about how important it is for everyone who hasn't made the decision to share data to think about how it will impact Glenda. I'm a person living with kidney disease. I have a transplant. Any drug that I take could have an adverse impact on me and my life. Any drug that I take could interact with some other drug and have an adverse impact on my life. I'm representative of a lot of people that are living with kidney disease. I'm representative of a lot of people who don't have kidney disease, but who might be impacted by drug-induced kidney injury. And to the extent that we could determine earlier who's impacted and if they are at risk for drug-induced kidney disease, it would be better for all of us in our community. So I just want to ask all of you that haven't made the decision to share data, don't think about it in terms of what's in it for you. Think about it in terms of what's in it for me, Glenda, the patient. Thank you.
Oh, wonderful, Glenda. Thank you. Well, I thought about how I was going to close out today for quite some time and was back and forth on going through something elaborate, and finally decided that I would keep it relatively simple. And so if we'll go to the next slide. Yes, so what I wanted to do is address a collaboration to address the action plan. So let's go to the next slide. And so this is the call to action, to advance data sharing to permit broad implementation of these novel kidney injury biomarkers. First and foremost, complete building the framework and the action plan. That's going to be very important for advancing any of these pieces. And it's going to require components of what I listed below here. Form a core group to advance understanding and implementation of these biomarkers into drug development first. Continue to solicit contributions from many of the sources that we identified today and yesterday. Everyone here, return to your organization and determine where there are applicable data sets that you can share. Come back to us and we can work with you on how to approach your team on how to share this data. And then as well, once we've established this action plan and executing on that, go back to that first slide that I had up. We're not going to physically, but go back to that first slide that I had up. He could stay here. Go to the current slides, excuse me. I meant metaphorically. Is beyond just looking at drug-induced kidney injury biomarkers, expanding these biomarkers into a broader context in the future. Once we've hit on and succeed here, we'll begin to expand into the other areas we identified in terms of efficacy endpoints in therapies for AKI, and also looking at chronic kidney disease. So that's the little bit of the aspirational component, but right now focusing on those first components that I brought up on the slide. And so with that, I would like to go to the final slide. I thank everyone immensely for joining us over 2 days. 5 hours twice is a lot of a webinar to join and participate and be engaged for this amount of time. So I appreciate everyone who's been here. Again, thank you to the Cpath staff for your work in the background to get this up and going and continuing it through despite the roadblocks we had that the pandemic put up. to all of the patients who participated and recorded videos and participated in the monthly meetings, as well as those in academia and industry who have been attending these weekly meetings, and to the rest of the audience and stakeholders who joined us today and provided additional perspectives. So with that, we will conclude today's meeting, and I hope you enjoy the rest of your week and are able to have a nice Memorial Day weekend. Thank you.